Dr. Sethi, R.S. Sethi, Head Department of Biotechnology, College of Biotechnology. Again, Gadwasu we call is the Guru Angad Dev Veterinary and Animal Sciences University, India. Dr. Sethi, could you please interact for a few seconds?
Good morning. Good morning, George, and thank you very much for accepting invitation and to be with us. We are very much excited, and thank you very much.
Yeah, and
you're welcome. It's a pleasure.
Yeah, thank you very much. And our coordinators, the one on pink turban, that is Dr. Prabjit Singh, my colleague, assistant professor in College of Fisheries. We all from
Good afternoon, Dr. George. We welcome you, and we are thankful for accepting our invitation.
Thank you. Thank you.
And Dr. Shiv Prakash, former Director of Extension Education and former Dean, College of Fisheries, Karnataka Fisheries College. Dr. Shiv Prakash, please, can you interact for a couple of seconds? Dr. Shiv Prakash, could you please unmute? Okay, anyway, he'll come back after some time. So I will introduce him again, okay? And you know, I don't have to introduce Dr. Prabhu Gowda. He's your own student, okay? So anyway, he will give introduction about you. So because he's the right person to introduce you, okay? And we have Naveen Kumar BT, our colleague. He's working in our same college, College of Fisheries. Naveen, could you please speak a few words?
Dr. George, good morning. I hope you remember we have met during Prabhu Gowda's marriage.
Oh, that's a great introduction. Yeah. So, and Dr. Anush, have you joined?
Yeah.
Yeah, Dr. Anush Tyagi, my colleague in College of Fisheries. That's Dr. George. Okay, brief introduction.
Good afternoon, Dr. George. Thank you.
Thank you. And once we start, then we'll have a formal introduction and welcome speech by Dr. Meera D. Ansal. With everybody's consent, I may start. Please, hosts and organizers.
Yeah, we can go ahead.
Yeah, Dr. Meera?
Yes,
okay.
Yes, you can start.
Okay, we can start. Dr. George, are you ready now?
Yes, whenever you are.
Fantastic, that's great, wonderful start. So,
what about Dr. Azad?
Dr. Ajay, he will contact soon. Okay, it's fine. Okay. He still has time. Okay. During our webinar, one of us will contact. Dr. Shiv Prakash is also there, so there is no problem. Okay. Thank you very much for a brief introduction about ourselves. Hope you are equipped with to start our international webinar, and it's going to be interesting, I guess. So let's hope for the best. At the outset, I take this opportunity to welcome Dr. Meera D. Ansal. Hope participants are fine. If you have any hiccups in between, please drop message on chat box so that we can try to rectify any problems if you face. So with that, I request Dr. Meera D. Ansal, Head of the Department, College of Fisheries, Guru Angad Dev Veterinary and Animal Sciences University, Ludhiana, Punjab, India. Dr. Meera, you are going to be live now, and please unmute yourself and start your introduction and followed by welcome speech. Thank you very much. Yeah.
Thank you, Dr. Shantanu. First of all, very good afternoon and good morning to everybody who has joined this webinar. Hope everybody is staying safe and doing fine. So I welcome you all to this international webinar on current advances in epigenetics and vaccine development in aquaculture, which has been organized to discuss recent developments and scope of epigenetics and vaccine development in aquaculture in context to India. So as we know, food and nutritional security are two prime, you can say, global issues, especially for India, having, you can say, over 1.3 billion people to feed. And if we want to have sustained production targets, we have to focus on some ecosystem approaches which are safe for the environment as well as for the consumer. And if we talk about aquaculture or fishery sector, we all know that it is the fastest growing sectors globally, and they are contributing significantly towards food and nutritional security at regional and global levels. So if we want to develop this sector sustainably, we need some safe innovations some safe innovations are required. And for this purpose, we need to strengthen our R&D programs through national and international linkages. So this is a very good gesture by Dr. Shantanu to start this webinar. So I congratulate you and bless the kids who have shown this lamp during this inauguration. And I continue with my words. In the recent past, as we all know in India, the government has initiated many academic programs for capacity building of students and faculties. And they are motivating the students and scientists from fishery background or from aquaculture to go abroad, work in foreign labs in developed countries so that they can learn new techniques. So it is, there is no doubt about it that we have to strengthen our linkages at national and international levels. So in this direction, we have taken this initiative that we want to strengthen our international linkages. So this webinar has been designed, or you can say conceived. And we have two eminent international scientists with us who will be sharing their experiences and ideas about application of epigenetics and vaccine development in aquaculture. So first of all, I take this opportunity to welcome our speaker, that is Dr. George Fernandez, Professor in Genomics and Molecular Biology from Nord University, Norway. So, sir, we welcome you on behalf of College of Fisheries, Guru Angad Dev Veterinary and Animal Sciences University. From this family, I welcome you into this international webinar. And I think Dr. Azad is still to join this.
Yeah, he'll join.
He has joined.
Azad is asking for link. Sorry.
Yeah, I will send it.
Okay, thank you.
Okay.
So Naveen, you send it. Naveen.
He's waiting to join, or he has joined?
He is asking for link somehow.
Okay, I sent him. Yeah.
Okay, so whenever he joins us, we will be welcoming him also. So never mind. So after that, for the speakers and all those participants who have joined us for the first time, I will give a brief introduction about our university, our college, and its activities. So College of Fisheries, Guru Angad Dev Veterinary and Animal Sciences University in the state of Punjab of India. It was established in the year of 2008 with a triple mission. The first mission was, or you can say is, to generate skilled human resource or professional human resource to cater the sector in all professional capacities. Second motive is to generate suitable technologies. Third motive is to transfer those technologies to the stakeholders for sustainable development of the sector. So if we talk about College of Fisheries in Punjab, we have 4 departments: Department of Aquaculture, Department of Fisheries Resource Management, Department of Aquatic Environment, and Department of Harvest and Post-Harvest Department. So we are offering various academic programs, which include UG program, it is called BFSC. Then we are also offering PG programs and PhD degree programs. And our major research focus is to enhance aquaculture productivity and sustainability through innovations involving diversification, nutritional management, health management, disease diagnostics, surveillance programs, organic concepts, food safety concepts. And we also have a dedicated team who is working on ornamental fisheries. And our fishery resource management people, they are working on the natural resources to see or to assess the ecological health of our natural water resources like waters, wetlands, reservoirs, whatever we have in our state. And they are also generating database for the policymakers and planners. And if we talk about our processing section, they are working on the concept of zero wastage processing technology. See, if we see the changing environment, or we talk about global warming, so we have to develop climate smart technologies. So it will not serve the purpose if we just focus on climate smart technologies. We have to ensure that whatever food is being produced, whether it is from agriculture, livestock, or fisheries, whatever is being produced, every gram of that food is used in one or the other form. So our post-harvest people, they are working hard to see that how they can be can develop value-added products, functional products, and how they can utilize the processing waste into some other, in preparing some other valuable byproducts. So this is all about, or you can say the outline about the programs of College of Fisheries and all the departments, what type of work they are doing. But we want to improve our R&D programs. For this purpose only, we want to have linkages with eminent scientists at national and international level. So we are blessed, we are honored that at this moment we are having very two eminent scientists with us who will be sharing their experiences in two of very innovative areas, that is epigenetics and vaccine development in aquaculture. With this, I also take the opportunity to, you can say, not welcome at this moment because they have not joined, but I would like to thank our chief patron, that is Dr. JPS Gill, who is Director Research of our university, and our patron, Dr. KS Sandhu, who is Dean, College of Fisheries. They have provided all the support, all the inputs in organizing and conceiving this international webinar. At the same time, I am very much grateful to Dr. SM Shivaprakash. I welcome you, sir, to this webinar. Your collaboration is very delightful and your inputs are very valuable. So we are very much happy to have you with us on this webinar.
It's my pleasure. Thank you.
Thank you, sir. Welcome, sir. And our team of coordinators, as Dr. Shantanu said, that we have with us Dr. Prabhu Goda from Norway. And he has played an instrumental role in making communications with Dr. George Fernandez and to participate in this webinar. So we are very much thankful to you, sir, and we welcome you. And we are very much happy and honored to have you as part of our coordinators. So I welcome you to this webinar, and I welcome all the coordinators. Dr. Shantanu is there, Dr. Prabjeet, Dr. Anuj, Dr. Nameen, Dr. Jitendra. So you have made a commendable job. I welcome you all, and I'm thankful to you. And I know that I think Dr. Vineet Inderkaur has joined also.
Yeah, she has joined.
Okay, she has joined. So she's, I would like to introduce, she's head Department of Aquatic Environment, having another charge of Department of Harvest and Post-Harvest Technology. So she's also playing an instrumental role in, you can say, all the R&D plannings of these departments. So I welcome you, ma'am, to this webinar. At last, I welcome all the participants, either students or teachers, scientists, faculty from India and abroad who have joined us today in this webinar. And I hope that this webinar will be a grand success. It will be very much informative and interactive. And after this session, all the students, all the faculty members, they will be having enhanced vision, and they will be able to use, you can say, plan better things for the welfare of the fishery sector. With these words, I thank you all and welcome you all to this webinar. And with best wishes, I request Dr. Shantanu to proceed further.
Yeah, thank you very much for a wonderful introduction and welcome speech. I'm very gratitude for you. And as you already told, it's a wonderful gesture by lighting the lamp. On behalf of all the organizers, participants, we are lighted. It signifies it brings positive vibes. Because of light, we are on this beautiful earth. Let us bring positivity amongst all the people. And it is another thing is it's a exchange of knowledge, or in this case, transfer of knowledge. Dr. George and Dr. Ajad, they are going to share their expertise. Thank you very much. Wonderful kids they did. Thank you very much.
Yes.
So with this, I would like to invite Dr. Prabhugoda S to introduce Dr. George Fernandez. Actually, I received a CV of Dr. George. Then I thought, I'm not the relevant person to introduce. So Prabhugoda knows much better than anybody else in this team. So he's a right person to introduce George Fernandez because Dr. Prabhu has worked, he did his PhD, pursued his PhD, and also did his postdoc job with George. So he can introduce George to us and to our participants. Thank you, Prabhu, and please unmute, and you are going to be live now. Thank you.
Okay, thank you. I hear me, right?
Yeah, you're audible. Very clear.
Okay. Hello, everyone. Myself, Prabhu. I'm working as a researcher in Nord universities. I'm so honored and also so excited to be here and having the opportunity to introduce one of the leading experts in the aquaculture genomics, and also the one who introduced me to the world of epigenetics is none other than Professor George Fernandez. He's my boss. I did my PhD and postdoc with him, and he worked with so many fish species, and he has so wide knowledge. Maybe he will explain himself during his presentation. Moreover, just I would try to give only brief idea about epigenetics. It is an emerging field of science, particularly in the context of aquaculture. It's very important, and moreover, in India, there is no much especially in the selective breeding programs, the knowledge of epigenetics is very important. So, hope you all understand, and also you are so excited to be here from knowledge from George. So, on behalf of all the participants and also the organizer, I warmly welcome Professor George for his presentation. George, please go ahead with your presentation.
Yeah, thank you, Prabhu. George, you're going to be live. Please unmute and you can take over now. Thank you, George.
I will try to share my screen.
Yeah.
Yes.
Okay, can you see my screen?
Yes, fantastic.
Yes, George.
Okay. So good afternoon, ladies and gentlemen. I would like to start by thanking the College of Fisheries and the coordinators of this webinar for their kind invitation. It will be a pleasure to tell you a little bit about my research in epigenetics on aquaculture species. What I'm going to tell you, it's mostly fundamental research, so I hope it is not too scary, but it has a lot to do with the strategy of your college, like as presented by Dr. Mira. So it has a lot to do with the sustainability of the aquaculture sector, especially in this context of global warming and lack of resources like water. So we are developing this recirculating aquaculture systems, lack of agricultural products, lack of fish meal and fish oil. So we are actually replacing fish meal and fish oil with some plant ingredients. All these are related to epigenetics, because epigenetics is one of the ways, perhaps the main way, how the organisms integrate this information from the environment and try to adjust their metabolism, their physiology, their growth, their resistance to disease, for example, to produce a phenotype. Okay, so thank you very much, Prabhu, for your introduction. I had made my own slide with a little bit more detail about my background, since probably most of you do not know me. So I'm originally from, I will just switch on the pointer. I'm originally from Portugal. Well, actually, I'm originally from Mozambique, but I grew up in Portugal since I was 3 years old. In Portugal, in Lisbon, I did a degree in chemical engineering and biotechnology, which I finished in '98. And then I started this PhD program still in Portugal. It's quite a prestigious program by the Gulbenkian Science Institute, and it was a PhD in biology and medicine. So, but because of my background in engineering, I want to do something a bit more practical. So I found this lab in Scotland, Dr. Valerie Smith, who unfortunately passed away recently. And she was an expert in crustacean immunology, and she was starting a new line on research on antimicrobial peptides. So I thought, okay, this is very interesting to me because it has to do with biochemistry and molecular biology, and it's also in a field that has some clear application because these peptides, they are basically alternative antibiotics. They are natural antibiotics. So I did my PhD there, where I was looking for antibacterial peptides in the mucosal surfaces of rainbow trout. And I found quite a lot. Unfortunately, I just kept finding more and more, so I didn't actually have time to try to develop their application in the aquaculture sector. But at the time, several companies were very interested in trying this antibiotics, either being delivered through the feed or through these mini osmotic pumps and so on, because they have very interesting properties. I will not tell you much about, not the point of this talk, but they are very potent and they are resistant to proteolytic degradation and so on. So they're very interesting things. But after this, so my background is actually in, my doctorate was in fish immunology and comparative immunology. But then I started to become more and more interested in molecular biology, and this opportunity came up to do a postdoc in a leading laboratory still in Scotland in muscle development. This was Professor Ian Johnston's laboratory. And I stayed there until 2007, looking at how the environment affects the muscle growth in fish and what would be the genes that may be responsible for this. And then in 2007, I continued to move north. So I moved up here, very close to the city where Santa Claus lives in Finland, but this is in Norway there, next to some very beautiful islands called Lofoten Archipelago. And this is a picture of our university. This is the library here. Where I took a position as a professor in genomics and molecular biology. And I continued both lines of research in muscle growth and in innate immunity in fish. So my areas of competence are mostly fish immunology, especially innate immunity, and fish growth and muscle development. But perhaps since 2008-9, I started to become more and more interested in epigenetics in relation to growth and immunology, because I wanted to know not just what genes are responsible for this phenotypic plasticity of growth or production of antimicrobial peptides or other innate immune mechanisms, a bit more about the mechanisms above that can regulate actually the genes and the gene expression in relation to environmental factors like temperature, light, nutrition. And in this case, I will tell you more about this project on domestication. Domestication is very important in aquaculture, and it's still at a relatively early stage in fish compared to land animals. So it is something that I think it's really worth investigating, and now is the time to do that. And then, not on the side, because I also related to the other three things, three topics. I'm interested in how organisms adapt to different environmental conditions, how the genes evolve from a molecular perspective, and how these small non-coding RNAs like microRNAs impact muscle growth and how they are involved in the immune response in fish. Okay, so this talk will be divided in 4 sections. First, I will tell you a little bit about muscle growth for those of you who are not so familiar with it. And then there will be a brief introduction to epigenetics. And finally, I will give you 2 examples, one in Senegalese sole about the epigenetic mechanisms that underlie the plasticity of growth in Senegalese sole. And I will finish with an example of some results that I got from this project on tilapia domestication, where we are looking at epigenetic changes that may be related to growth in Nile tilapia. Okay. So, muscle is a very interesting tissue, very complex, like what you can see here in this picture. The structure of the cells is almost crystalline. Everything is so densely packed inside, there's almost no space for anything. And we've got these actin and myosin fibers that just slide across each other and generate force and are responsible for movement. They can generate heat and so on. It's a very, very complex tissue. It has a lot of nuclei, you can see here. So muscle is a polynucleated tissue. So we don't have individual cells with one nuclei. We have muscle fibers that have many nuclei. And because everything is so packed, these nuclei tend to be pushed to the side. And I'm not talking about cardiac muscle, that is a bit different. I'm talking about skeletal muscle, which is the muscle that makes most of our body and most of the body of the fish as well. And it makes up anything between 40 and 60, 70% of the whole body of the fish. So for aquaculture, it is very, very important. It is the most of what we eat from a fish is muscle. And it all starts very early during development. What we can see here in this picture on the left is a Japanese pufferfish embryo that is going through a stage called somatogenesis. It's called somatogenesis because it is a stage that corresponds to the formation of these chevron-shaped structures on either side of the notochord. This is the notochord here in whitish in the middle. And then we have the different somites. Each of these is a somite. And it is, pardon, it is in the somites that come from the paraxial mesoderm that all the skeletal muscle will be formed. I'm not going to go into a lot of details, but there are many, many genes that are involved in this, including some muscle-specific microRNAs that I will tell you in a minute, these myogenic regulatory factors, MIF5, MIF6, myogenin and MyoD. They are very, very important in muscle development. And then several genes involved in the signaling pathways like the mTOR, AKT pathway, and the MAP kinase pathway, and so on. So there are literally hundreds of genes that are involved in muscle growth. It's very, very complex. And the muscle fibers themselves, they are derived from these myoblast progenitor cells that they can either proliferate to generate more myoblast progenitor cells, or they can become a little bit more committed to the formation of muscle fibers, and they can produce these myoblasts. So these ones are less of a stem cell than the myoblasts, so that they have to differentiate into muscle fibers. So these myoblasts, they will then go to the places where we need the muscle, so they'll migrate, and then they can either fuse with each other to form a new muscle fiber, like we can see here, new myotube, or they can just basically donate their nucleus to a growing muscle fiber, a growing myotube. So now this myotube will have 4 nuclei, so it can actually become bigger. And then it will differentiate, like starting to express this actin, myosin proteins and so on, so muscle-specific proteins and so on. So this process here in the bottom, it is called hyperplasia, which is the formation of new fibers. And this process here is called hypertrophy, which is the growth of existing fibers, okay? So in mammals, we are born with pretty much the same number of fibers that we will have in our adult life. So a baby has almost the same number of fibers as you have. The difference is that they are much smaller. Okay, so what happens after birth is that the fibers just grow. There are a few exceptions. So we can form new fibers. For example, if there is an injury, to some extent, the muscle can regenerate and form new fibers. But in general, our fibers grow by hypertrophy. So it is this process here that happens in mammals after birth that is responsible for our growth. In the case of fish, both these processes happen throughout most of the life of the fish. Okay, and this is one of the big differences between muscle growth in mammals and muscle growth in fish. There are many, many similarities. Most of these genes are the same and so on, but there are a few features of fish muscle that are very special. And one of them is this one that I have just told you, that both hyperplasia and hypertrophy contribute to growth after hatching in the case of the fish, unlike mammals, okay? And this is because if we think about it, well, the fish, when they are born from these eggs, they're very, very small. You know, they can be anything from just a few milligrams, let's say like a tuna egg. And then this fish can actually grow to several hundreds of kilos. So we are talking about the growth of 10,000 times, 100,000 times. So this would be the equivalent of us becoming the size of whales. And to be able to do this, it's not enough that the fibers just grow. This would cause a lot of problems with the diffusion of nutrients and oxygen to the muscle fibers and so on. So the fish actually need to make more fibers to grow this much, okay? And the muscle in fish, the two types of muscle, of skeletal muscle, slow and fast muscle. So the fish have this slow muscle that's a bit more reddish. You've probably seen it when if you eat fish. And they use this muscle for sustained swimming. This is the muscle that they use normally, or mostly. And then they have the fast muscle, which is the white muscle that makes most of the body of the fish. And that is used for this bursts of speed, like if the fish wants to escape a predator, or it really has this opportunity of grabbing some food that may run away or swim away, they use the fast muscle for these bursts of speed. And in the case of fish, these two types of muscle fibers, fast and slow, they are organized in anatomically separate layers, unlike mammals. So this is a very big advantage if you want to study only fast muscle or only slow muscle. It is pretty tricky in mammals. There are a few muscles that are more rich in one or the other type of fiber, but in general they are mixed. Whereas in fish, it's very easy to find regions of the fish body where you only have fast muscle or you only have slow muscle. Okay, and finally, I've got it here that the muscle growth shows plasticity. By this I mean that growth can change a lot, not only during the very early stages of the...
Dr. George, can I interrupt
Yes.
for a couple of seconds?
Sure.
Just I wanted to know, just a small interruption. It's for participants. We are having problem with the chat box. You can drop your messages in WhatsApp groups. If you have any problems, please let us know. Okay. We cannot see your information or messages on chat box. There is some technical error. If you have any problems, please call us or drop messages on WhatsApp group. Thank you very much, and apologies for interruption, Dr. George. Thank you.
No problem.
Yeah, thank you.
So I was saying that the muscle growth shows plasticity, and this means that it is not fixed. It is not determined. It is plastic. It can change. It can be modulated by the environment. It can be modulated by how much the fish eat, what type of diet, if by their immune status, if they are sick or not, by the presence of parasites, by temperature, by light, by many factors. And from the lab where I did my postdoc, they had some really excellent examples. These were one of the first examples of plasticity of muscle growth, and I've chosen this one. It is an Atlantic salmon, and basically they took the embryos from 360 families of salmon. So this really excludes genetic effects. And the other incubated these eggs at the normal temperature, which is called cold ambient temperature, or in water that was heated by just 1 to 2 to 3 degrees. So you can see it's a very small temperature difference. Like if you think about global warming, 1 degree Celsius in 10 years, 20 years is kind of expected. So this can actually have an effect on muscle growth, as they've shown in this example. So after the fish were tagged, so they were at different temperatures for a very short time until they were tagged, and then they were put together and grown in the same sea cage for 3 years, almost 3 years, so 900 days. And we can see that even after 900, so these fish that are represented here by some black squares or some open circles, they actually spent most of their lives in the same conditions. The only differences were here really at the very beginning of their life. And we can still see huge differences in fiber number. This was about 20% difference between the two groups. It was 20% higher in the fish that came from the cold temperature group. And this is really amazing. It's amazing that just 1 to 3 degrees can have an effect of 20% in the fiber number, which would reflect in 20% of the weight, let's say, not exactly, but around that. And also that this can persist for so long, okay? It is really amazing. And this, when I was in this lab, then I started to think about epigenetics. Okay. And so what is epigenetics? Well, epigenetics, it's a term that was coined by Waddington in the 1940s to explain something that he was not really sure about, that was something that was beyond or above genetics, when he was trying to explain cell differentiation. You know that we have many different cells in our body, and but the DNA of all these cells is pretty much the same. There are a few exceptions, like the immune cells, like the B cells, for example, or the gametes, but most of our cells, they have the same DNA, you know, like a skin cell or a hair cell, they have pretty much the same DNA. And he was interested to find out why do we have cells with the same DNA that become a hair cell or a hair or a cell of your skin. So he came up with this concept of epigenetics and the epigenetic landscape and so on. I will also not tell you much more about that, but nowadays, probably the most common definition of epigenetics is this one, which is the inheritance of traits and changes in gene expression that don't necessarily need changes in DNA sequence. So we can change the expression of the genes, the phenotype, and this can even be inherited without changing the DNA sequence. Okay, I normally, when I'm lecturing, I normally compare epigenetic mechanisms, epigenetic marks, to light switches or punctuation marks in a text. Okay, so these very small punctuation signs like commas or full stops or semicolons and so on, they determine when and how the genes are turned on, and this will have different outcomes in the same way that they can affect the meaning of a sentence, these punctuation marks. And a nice example is when we look at this sentence, let's eat grandma. Most people would think that we are just asking grandma to have some food with us, right? But if I remove this comma, then it becomes let's eat grandma, which has a completely different meaning. And the only difference between this and this is just the comma, okay? So these epigenetic mechanisms, this is just to illustrate that they are very small changes, but they can have a huge effect. They can completely change the meaning or the function of a gene by switching it off or activating it. And therefore they will affect the phenotype dramatically. This is an example in mammals where you can see two mice that they're actually identical twins. So they have exactly the same DNA. But just by looking at them, you can see that they're really, you would never guess that they are twins because their size is completely different. This is much, much bigger. And also the color of their coats is completely different. And this is controlled or regulated by epigenetic, one type of epigenetic change in one gene. This gene called obese or agouti. And this gene can actually, or these epigenetic changes can be modulated by environment and nutrition. Unfortunately, we don't have these nice examples in fish so far. That's why I showed you this one in mice. But this phenotype, the yellow fat mouse that is prone to cancer and prone to diabetes, and also has learning problems or difficulties, it can be regulated by nutrition. So if you put this, when this mouse that will become fat and yellow and with this diabetes and cancer conditions, if the mother mouse has a nice food that is rich in these methyl donors, like folate, for example, then instead of being born a yellow mouse, it will actually be a normal mouse with the normal size and the brown coat. So this is amazing as well, like how the diet of the mother can actually affect the phenotype of the offspring, and something that will actually persist until the end of their lives, because this yellow mouse, it will continue to be yellow and fat and will have diabetes and will die earlier and so on, just based on the small changes like these methyl groups in one single gene. Okay, so how does this epigenetic mechanisms work? So there are different types of mechanisms. The main ones are the modifications of DNA. And here it's an example of DNA methylation, but basically just like small changes in the DNA, not changes in the sequence, but just things like methylation or hydroxymethylation that will affect the expression of the genes. You can also have modifications of the histones. The histones are these proteins that are wrapped around the DNA very tightly. And in order for the genes to be expressed, this needs to be loosened up a bit. And some of these modifications, like let's say acetylation, in general, it will open up the histone and allow the gene to be transcribed. So this is the regulation of the chromatin structure by this histone, and histone modification is a very important epigenetic mechanism. And finally, we have these RNA-based mechanisms that include different types of non-coding RNAs, like long non-coding RNAs and microRNAs. I will tell you more about microRNAs. Pardon. Okay, so DNA methylation is basically the addition of a methyl group, a CH3 group, to cytosines in the DNA. Here they're represented by these red circles. And the enzymes that do this are these DNA methyltransferases. They can put or remove these methyl groups. And normally when the gene has these methyl groups, these yellow dots in regions near the promoter, near the start of the transcription of the gene, then it will not be expressed. It's not always the case, but in general, DNA methylation in regions near the promoter or within the promoter of the gene will repress the expression of that gene, okay? So this is very important. It's probably the most well-studied epigenetic mechanism, this DNA methylation. Okay, another example are the microRNAs. These are, oh, this was a video, but I don't think it is working. Maybe if I remove the pointer, will I be able to? Yeah. So microRNAs, they are made in the nucleus by RNA polymerase II, and they have this this precursor structure, this pri-microRNA, that is then cleaved by DROSHA and the DGCR8 to form this pre-microRNA that is then exported out of the nucleus by exportin 5, and then it is included in this complex and cut by dicer to produce the mature microRNA that will then be incorporated into this Argonaut complex, and it will bind its target. So basically, it finds a gene that has a complementary sequence, and it will bind to it, and then it will inhibit its translation. If the complementarity is almost perfect, it will actually induce a degradation of that transcript. And so basically microRNAs are negative regulators of gene expression as well. So if you have a microRNA being expressed that targets a particular gene, then the expression of that gene will be downregulated, will be less. Okay. And sorry, no, I need to get this pointer again. Okay, and their involvement in epigenetics can be direct or indirect. So they can actually regulate the expression of a lot of other genes involved in epigenetic regulations, like these histone methyltransferases that are responsible for adding these methyl groups in the histones. They can also affect the expression of these DNA methyltransferases and therefore affect DNA methylation. Or they can have also a more direct action. So they're also involved in epigenetic regulation. Okay, so this morning I was checking how many publications we find in PubMed if we type epigenetics and aquaculture. And it is amazing that last year, I checked this last year, and it was about 90 something. So I was expecting many, many more this year, but no, only 128 publications. This is really very, very little. And we can see that if we go before 2009, we don't find anything. So this is really a new and emerging, it's still an emerging field, okay? And when it comes to, and even in models, fish species, there's very, very limited knowledge compared to mammals or plants. So when the work that I'm going to present to you started actually around 2008-9, and it has to do with the developmental plasticity of growth in Senegalese soil. Because by this point I was collaborating with a group in Portugal, and they were trying to develop several aspects, or to improve the farming of Senegalese sole, and the growth was an issue. And since we knew that the embryonic temperature regulates growth in several fish species, I wanted to know how does this affect the growth in Senegalese sole, and if the microRNAs and these DNA methylation mechanisms are involved in this process. Okay, so the Senegalese sole is a marine species of very high economic value, especially in the south of Europe, especially in Portugal and Spain. It is currently farmed. Some of these problems have already been solved. At the time, the main problems were the control of captivity breeding. This is still a big issue. When you bring the fish from the wild and you put them in captivity, after a few years, the males produce very, very little sperm, if anything. The females are still fine. They still produce good quality eggs, but it is very difficult to get them to breed because of the males. And quite often you end up needing to get wild fish, which makes it impossible to develop a breeding program. But also growth, deformities, and susceptibility to disease were still problems as well at the time. But the focus of this was on growth and how we can improve growth by changing the developmental temperature. So we designed this experiment where we had a batch of eggs here that we split in two groups. And we placed one group at 15 degrees and another one at 21 degrees. And we just let them develop and took samples at different stages of development, like we can see here, 12 hours, 20 hours, 34 hours. That corresponds to 50% epibole, 20 somites, or when they were hatching. At 15 degrees, things happened a bit slower. So it was at 22, 36, and 62 days. And then at 14 and 15 days post-hatching, when they were kept at the same temperature. Okay, so we saw that there were differences in growth between these groups, and we wanted to see if the microRNAs that they were expressing were different. So in those days, we used still this CLC, a bio to analyze the data, and we sequenced, prepared the libraries, and then we sequenced them on this solid machine that we used to have. I don't know, maybe we still have it, but we cannot use it anymore because there are no consumables for this anymore. But anyway, it was quite a nice machine. It produced a huge sequencing output. And then we just did this bioinformatic analysis. And we found out that what you cannot really see, and apologies for the very small font, but here we have 15 degrees and 21 degrees at the first developmental stage, the 75% epibole. And then again, 15 and 21, 20 somites, and so on. But anyway, I'm not expecting you to see these differences in detail anyway. It's just for you to see that the patterns are changing between the different, well, both between the different temperatures and different stages. So these vertical columns. Okay, so we identified, for example, here is a very big difference in these first two columns. This is one developmental stage, so 15 and 21 at 75% epibole, compared to the rest. So this bright green means that there's very little, almost no expression. This bright red means there's very high levels of expression. So we can see that there are big differences with developmental stage and temperature. So we focus on a few microRNAs that were both changing with temperature or stage, and also that we knew that they're important for muscle growth. And we found a few, for example, this 26A, 130C, 181A, 206. They were expressed more in the group that was kept at 21 degrees than 15 degrees. So the higher the incubation temperature of the eggs, the higher the expression of these microRNAs, these 4 microRNAs that are involved in muscle. We then predicted the targets of these microRNAs to see which genes may be involved in this. And again, this is more complicated than it looked. If I wanted to explain this properly, it would take quite some time, but I just want you to notice that these microRNAs that were changing with temperature are very important for my genesis because they can affect the signaling pathway, this IGF and the IGF-1 receptor genes. which in turn can affect proliferation and differentiation in different ways. So they can inhibit or inhibit proliferation in this case, for example, and induce differentiation, or in this case, it could induce proliferation and inhibit differentiation and so on. So each of these microRNAs can have different targets. For example, this 130C can inhibit both MAP kinase 9 and this EIF4E with different effects on muscle growth. And one very interesting one was this 181A that affects calpain 1. This is a gene that is essential for the fusion of the myoblasts. So it will, if we have less calpain, we will have less fusion of myoblasts. So it will induce the proliferation of the myoblasts compared to their fusion. So anyway, we looked at this and we found out that this assembly of different expression levels of microRNAs, of these microRNAs, could explain the differences in growth that we saw between the two temperature groups. And then when we looked at the expression, at the methylation of the myogenin promoter in Seneglisol as well, we found something very interesting. This myogenin is one of the muscle regulatory factors. It was one of the first genes to be found that were known to be involved in muscle differentiation. It is a master transcription factor. Actually, if we take, for example, a different cell type, like a fibroblast, and we make them express myogenin, these cells will become muscle. So it's a very, very important transcription factor in myogenesis. And it is critical for muscle for differentiation. Okay. So to look at this, we designed a similar but a bit different experiment. So this time we actually had the embryos at the same temperature, 20 degrees, until hatching. And then after hatching, we put them at 3 different temperatures, 21, 18, or 15. And we just let them grow until metamorphosis. You know, the Seneglisol is a flat fish, so until the point where the eye rotate and they become completely flat. And this happened again at different times, depending on the temperature. But then we looked at how big they were and also how the muscle structure, the muscle fiber number was a bit in the 3 different temperature groups, okay? One thing that we can note is that the fish that were reared at 21 or 18, they were much bigger. So like they weighed almost 2 times more, sorry, this is here. At the metamorphosis stage, they weighed almost 2 times more at 21 than they did at 15. And this difference was less, but still persisted even after metamorphosis. But we focused mostly on this because that's where we found the biggest differences in size between the temperature groups. Also, we looked at the muscle cellularity, and the number of fibers was different. Both the number that we can see, sorry, this is both the diameter and the area of the fibers were different and were higher at 21 than at 15. The number was not that different, not significantly different. So this suggests that there is more hypertrophy going on. So the fibers are becoming bigger rather than increasing in number. When you look at the expression of some key genes like myogenin and these DNA methyltransferases that put these methyl groups in the DNA, we found that at 21, which is the pinkish bars, there was higher expression levels, almost 2-fold higher in the fish reared at 21 compared to 15. And on the other hand, the expression of some of these DNMTs, like DNMT3B and DNMT1, it was higher at 15 than at 21, suggesting like an overall higher expression. Not necessarily, but it indicates that there may be a higher expression of DNMT1 and DNMT3B at 15 compared to 21. Okay, so there could be more methylation at 15 than at 21. Of course, we didn't measure enzyme activities or even protein levels, so we don't really know exactly, but this is what the gene expression results suggest. Okay, this is sequence alignment of the promoter region of the myogenin gene in different fish species. I'm not going to bother with this, but these boxes here with the colors, they are very important regions in the promoter of this gene. So because these are the places where these transcription factors like MEF3 and so on, they will bind here, and they have to bind here if the gene is going to be expressed. If they cannot bind for some reason, then nothing will happen. Okay, and we found that in some regions, for example, at this position -203 and this position -72, there were significant differences in the methylation, so this cytosine methylation between the temperature groups. So in the fish that were reared at 21, which again is this orangey pinkish color, there was less methylation in this position compared to 15 or 18. And the same at this position -72. So less methylation and more methylation at 15 or 18. So this again indicates less methylation, indicates more gene expression. And we summarize this in this picture that the fish reared at 15, they are smaller than the fish reared at 21, probably because they express more DNMTs and the myogenin promoter is more methylated, and because the myogenin promoter is more methylated, there is less expression of myogenin. The opposite is happening at 21, okay? Pardon. So just to summarize this Senegalese soul study, we found that there were several microRNAs differentially expressed with the developmental stage and or with incubation temperature, and this was correlated with growth. Temperature had a long-term effect on the expression of microRNA, so even if the fish after hatching were kept at the same temperature in common garden, we still saw differences in microRNA expression patterns, suggesting that this temperature effect may have some kind of persistence. And also the second part of this study provided the first evidence that DNA methylation may be one of the mechanisms responsible for this thermal plasticity of muscle growth that we normally see in fish. Okay, so in the second part of my talk, I will give you a brief presentation of a project that I got from the European Research Council to look at the domestication of Nile tilapia. And this is a very big project, and I will only tell you a very, very small fraction of the results regarding a new type of DNA modification that is called DNA hydroxymethylation. Okay, so the idea for this project came from the huge gains that we can obtain by selective breeding during domestication of, in this case, it's a Nile tilapia, this example that we see here. We can get an increase in growth compared to the wild fish by almost 85% just by picking the biggest fish in one generation as parents for the next generation, and so on. Okay, and being originally a chemical engineer, this for me was really amazing. I was very much amazed, and then I was thinking, okay, how can this happen so quickly? You know, in just one generation, to get a weight gain of about 15%. And I was thinking, okay, this cannot just be genetic. So then I was thinking about these epigenetic mechanisms that may be playing a role in this. And again, when I started this project, there was nothing known about the role of epigenetics in fish domestication. So I decided to go for this mini selective breeding program at our university, where we were going to collect fish, establish this base population, and then develop two lines, one selected line of fish that grew better than the average control line. Okay, so first we went to Egypt, like very, very south this is the River Nile that goes up. And we were here near Luxor, very, very nice place, like you can see here, you know, famous for the pyramids and the tombs that we get on the other side of the river, of the place where we were fishing. And we were catching this Nile tilapia with traditional traps like this, or with gill nets that they would put around. And then this is also a video, but I'm not going to show it because I don't have much time, where they made a lot of noise, and then the fish would just be scared against the gill nets and we'd just jump in the water and pick them up. And then we had this makeshift laboratory in the hotel, a room in Egypt, and then we transported the fish to our research station in Buda. These are our first babies from 2016. They are now huge. I don't know exactly how many kilos they are, but they are probably around 3-4 kilos, the fish from the F0 generation, at least, maybe more. And we have 3 generations. The example that I'm going to show you has to do with some fish that we found at the very beginning. They are not the wild fish, but they're the first generation that was born in captivity, that we call base generation. So we noticed that among these fish, there was a huge variation in size in some families. Okay, not others, but in some families. Like we can see here, these are the weights of several individuals that they come from the same mother, the same hatchery conditions, and they are at the same age and reproductive status. So even these tiny females that were about 20 grams, some of them even less than 20 grams, they were already sexually mature, so they were full of eggs. But some weighed 20 grams, some weighed 90 grams. And in the case of the males, the difference was even bigger. Some fish were about 9 times bigger than their counterparts, even if they're brothers. So they have a very similar genetic background. They were kept in the same conditions, fed the same water temperature and so on, and huge differences in growth. So then we decided to look in a bit more depth into the differences in hydroxymethylation patterns between these 2 groups. Between the big males, the example I'm going to show you, between the big males and the small males that were about 9 times smaller. And at this time, nothing was known about this new epigenetic mark called hydroxymethylation in fish. This is somehow related to DNA methylation because it is during this demethylation cycle, hydroxymethylation and methylation, they can be sort of converted in each other through different enzymes. And it has been found quite some time ago, but people thought it was just sort of an intermediary compound in this formation of DNA methylation or the demethylation cycle. But by this point, there were some publications showing that this was actually a stable DNA modification in somatic tissues in mammals. And there were a couple of examples showing that this hydroxymethylation mark can also regulate transcription in a way similarly to DNA methylation, but normally having an opposite effect. So what we think is that when we have hydroxymethylation, we have an induction of transcription. When we have DNA methylation, we have a repression of transcription. So these 2 mechanisms, they kind of have opposite effects, but this also depends where in the genes they are occurring and about different other different facts. So it's not as simple as this, but the general picture is that hydroxymethylation induces transcription and methylation represses transcription. And at the time, and even now we know very little, but at the time nothing was known about hydroxymethylation and growth in fish, in any fish, even in zebrafish. So we decided to look at this in our tilapia, the big and small males. And we use this technique called reduced representation hydroxymethylation profiling that enables us to look at hydroxymethylation throughout the whole genome of the fish. So we look at hundreds of thousands of positions throughout the genome. And then we made this map depending on where. So each of these parts of the circle represents a chromosome or a linkage group that's related to a chromosome. And the height of these bars represents the hydroxymethylation level. So if you have a bar like that, very high, it means that in this position, there is 100% hydroxymethylation in the big fish. And maybe, I'm guessing here, but let's say 30–40% in the small fish. So in many positions, the hydroxymethylation pattern was similar between big males and small males, but in many other positions, in many other genes, there were big differences, significant differences between the big males and the small males. Okay. When we looked at this, we had more than 250 genes that had significantly different hydroxymethylation levels between the small fish and the big fish. Sorry, the small fish are here on the right and the big fish. Most genes had higher hydroxymethylation levels in large fish, and they affected many relevant biological processes that are directly related to growth. You know, for example, muscle structure development was one of the gene ontology categories that were strongly affected by this DNA hydroxymethylation differences between groups, and also related to metabolism. For example, this deacylglycerol kinase activity or glycerolipid metabolic processes and so on. And there were even genes that were directly involved in regulation of muscle growth. Okay. It was just to show that we also found changes in hydroxymethylation in genes that are mitochondrial genes. This was something that is also very new, including that gene there. And so this can actually potentially affect energy metabolism and therefore muscle growth. Especially this diacylglycerol kinase was very interesting because we found that it was more hydroxymethylated in big fish. And the big fish, well, they grow more than the small fish. So if we have higher levels of this, then probably, or maybe, there is higher expression of this, and then maybe more activity of this, which would result in higher growth. Okay, this was just one simple example. So to summarize, the mapping, and this is actually the first mapping of hydroxymethylation in fish, showed that there were many differences in several genes in the hydroxymethylation levels of, in the hydroxymethylation levels of several genes in liver that could be associated with growth rate. Some of these differences were in the introns of genes that were directly involved in muscle growth, lipid and energy metabolism. And these results show for the first time that hydroxymethylation may be an epigenetic marker of growth in fish. And this is something that we are exploring now. And this is actually part of Dr. Prabhu Guda's postdoc. Okay, so where are we with epigenetics? Well, we are still at the very, very, very early stages. There are many problems or gaps of knowledge that need to be addressed. There are technical issues. It is not so easy to determine these epigenetic marks, and especially to analyze the data. We get huge amounts of data that takes a very long time to analyze. We know almost nothing about the inheritance of these epigenetic marks, and this is something that we need to know if we are going to use them as biomarkers. We also don't know how stable they are, if they will be reverted if we change the environmental conditions or not. And this is, again, something that we cannot use a marker if it is not stable. But I think that it is definitely worth looking into this because they have a huge potential to improve the profitability and sustainability of the aquaculture sector in various aspects. I just mentioned growth, but for example, disease resistance is another one, flesh quality is another one, and so on. So there are many, many aspects that show plasticity, environmental plasticity, and that could be epigenetic regulated. So I think that in the future, hopefully in the near future, we'll be able to develop epigenetic markers that will enable us to select for more robust fish that will contribute to a more sustainable aquaculture industry. Okay, and just before I leave you, I'd like to thank you all for your attention and to thank some of the people that were responsible for most of this work. So Catarina Campos was a PhD student that worked on the Senegalese Soul project. Yanis, Tom, they worked on the Epifish project, producing the, Yanis was responsible for the hydroxymethylation results. And Tom has done some DNA methylation work, and Prabhu is validating this DNA methylation and hydroxymethylation and microRNA markers. And I would like to thank all the other members of the Epifish team, and of course the institutions that provided the funding, especially the European Research Council, the Research Council of Norway, the Foundation for the Science and Technology in Portugal, and also my university. And thank you again for your attention.
Thank you very much, Dr. George, for your wonderful sharing the knowledge with us. It was a great honor to have with us today. And it's kind of, what you call, we can start making collaborations in what you call, in terms of aquaculture, and also we can start having student exchange programs, the developed institute. Already I understand that our Indian Council of Agriculture Research has taken a great step sending Indian students to Norway. Some of the universities in Norway, they are pursuing higher degrees in our institute with scholarship. So we are, congratulations to ICR, and we are very grateful to ICR. Just I request, let's have a kind of small interaction. We are receiving some questions about epigenetics in aquaculture. I request Prabhu Gowda and Anush Tyagi, two moderators here, so who are getting questions by the participants. So please get ready, and just for a few minutes. Before they start question, I got one question from a student from Nepal. So it's an interesting question. So I understand, I work back in Australia. I worked about an aromatase gene which controls sex in or gender in fish. Is there any role of epigenetics on controlling the sex or gender in fish? So basically we give some kind of hormones to change the gender of the fish to get the benefit. Is there any possibilities the epigenetics can play a role in controlling the sex of the fish or gender of the fish? Prabhu and Anush, please get ready for the next question. Sorry to keep you engaged with the questions. Okay. Yeah, please. Thank you.
It's nice to discuss science. That's very good. Yes, the answer to your question is yes. There are several examples. Actually, that has been better studied in growth, at least in aquaculture species. So, for example, in European sea bass, we know that the sex of the fish can be controlled to some extent by temperature.
Yes.
And the methylation of one gene, like this aromatase gene, has been shown very clearly. This was actually by Professor Francesc Piferrer, who's a collaborator and a friend, that the temperature induces different changes in the methylation status of this aromatase gene, which in turn will result in male or female European sea bass. And more recently, I think it was about 3 years ago, a very nice paper published by a Chinese group where they did this genome-wide mapping in a flounder species. And they also showed that, again, we can reverse the sex of the fish by temperature, and the underlying mechanism is an epigenetic mechanism related to DNA methylation.
Thank you very much. Before they take up, I need to introduce one more person here, Dr. Ambekar Eknath. He was a director in NACA in Southeast Asia, and also he worked in Norway for several years. And when he was in NACA, his contribution for developing gift tilapia, genetically improved farmed tilapia. Okay, I welcome, and if we can unmute and interact, that would be great, Dr. Eknath sir. Could you please come on live and interact with Dr. George? Before that, so I request Prabhu and Anush Tyagi to please come on for interaction session. Thank you.
I wanted to stay by myself. Hello, can you hear me?
Yes. Thank you very much for joining us, sir.
I just wanted you youngsters to interact. I just wanted to be a witness to this exciting interaction between you, the future generation. But anyway, thank you for recognizing my work, which was done over a period of 30 years.
Yes.
And I owe it a lot to a lot of people. You know, Norway, we were working with Nofima. It was earlier called Aquaculture.
Yeah.
And I have worked with them for more than 20 years, and we still work.
Oh,
and I really enjoyed the talk by George. If I may ask a few uninformed questions to George. George, hello?
Yes, yes.
You see, a lot of the early 19s, 20s, or 30s, a lot of work has been done, but it was not called epigenetics at that time. But it was mostly to change the sex of reptiles, for example, by temperature manipulation. And today you have given, with your group especially, you have given an underlying mechanism for such changes. It was just a fantastic presentation today. I really enjoyed it. But is epigenetic mechanisms, they are reversible, are they? When I say reversible, so the first generation, if I just allow them to grow to, let's say, embryo development from 15 degrees to 21 degrees, but the next generation, what happens to the progeny which comes out of this generation?
That's a very good question. And then unfortunately, it's a question that we still don't have a very good answer to, you see. So I think my personal belief is that some epigenetic modifications are permanent or more permanent, some are more reversible.
Okay.
Because, you know, epigenetics is very important for the adaptation of an organism to environmental conditions throughout the life of the organism, right? But it's also very important in evolution of a population, of an organism as a population. So I think that some changes will be, epigenetic changes will be more permanent, so they will be passed to the next generation, some others not. I can give you an example. I think this has been published. So I've got a collaboration with Dr. Kaya Sharvan from the Institute of Marine Research. It used to be called NIFES. Probably when you were in Norway, it was still NIFES. But 2 years ago, they got, they merged with the Institute of Marine Research. And she was coordinating this study, of which I was part, where they fed zebrafish with a diet that was very poor in 1C nutrients. And then there was an effect on growth that was expected. And they realized that after even 2 generations, there are changes in DNA methylation between groups. Even if in the 2nd generation, you feed the fish from the 2 groups, they are fed exactly the same. You know, their parents were fed different diets. The 1st generation offspring will be fed exactly the same, and you still see these differences. So some of them seem to be permanent, at least for 1 generation. I don't know how many generations this would keep, but then other changes, they will be reversible. And this is something that we really should put more research effort into, because if we want to apply this in agriculture, we need to know more about the inheritance of these epigenetic marks.
Just one last question with the permission of the hosts.
Oh, thank you, sir. Go ahead, sir, please.
George, just
Yes.
to get the definition of epigenetics, in the beginning itself you said epigenetics does not necessarily involve changes in the DNA sequences.
Mm-hmm.
Now, can we call epigenetic, a fish produced to epigenetic manipulation, a GMO?
That is, no, I don't think, well, at the moment, no.
Thank you very much. So,
I don't know.
Can we get around this problem of GMOs?
That is true, but you know, in 10 years, I don't know if the definition will become as strict as, okay, it will be a GMO if there is a epigenetic change in this particular gene. But so far, no. And that is a good thing. In Europe, we are very strict with the GMO regulations in aquaculture. So if we can make bigger fish or make fish that grow faster or better, or that are resistant to some disease without changing their DNA sequence, then it is really a big advantage.
Excellent, excellent. Thank you very much.
Thank
you very much, the hosts also.
Oh, thank you, sir. Thank you very much, sir. Yeah,
you for the
yes, it's our pleasure. And thank you, both of you, for a wonderful interaction. Thank you.
Okay,
and great scientists, both of you. Yeah, thank you. Now it's over to Prabhu and Anushtagi, please.
Yeah.
Yeah, please.
Maybe I will take the first question.
Yeah, thank you.
Yeah. Thank you, Dr. George, for your very insightful presentation. And I'm sure most of the people here will not be forgetting the epigenetics after your grandma example. So that's very good, especially for the students. So actually, one of the most frequent question we were getting in the group was about the inheritability of these epigenetic changes, which Eknath sir has already asked, and you have replied it, covered it. So I have one more question on behalf of participants. So basically, for these epigenetic changes, are you studying or targeting any specific genes, or you are targeting on the whole genome basis, like targeting some bisulfite sequencing and then selecting from those data, like these are the genes which are getting changes in the methylation patterns?
Nowadays, I'm using a genomic approach. So okay. I don't target any gene or modification in particular. So I look at how this modification looks across the whole genome, or is distributed across the whole genome. And I compare the profiles between two groups, let's say, of fish that grow faster compared to fish that grow slower. And then I will see where the differences are and if they are within any interesting gene. And sometimes we find very clear differences that are in genes that we didn't expect to be epigenetically regulated. And then you find out by going through the literature and so on that they actually have some connection to some other gene that we know is involved in muscle growth. So it makes it much more exciting this way. So we find a lot of unexpected connections between DNA modifications of some genes and growth. We also found some other ones that we cannot explain, of course, but makes it more interesting.
Thank you. Thank you. So I will hand over to Dr. Prabhu now.
Yeah, thank you. And we have time for only one more question by Prabhu. Yeah.
Maybe like Prabhu ask it.
Yeah.
Hello, George.
Hello.
I have I think so many questions, but anyway, I have got permission to ask one question.
Just for on a lighter note, don't ask the same question which you have asked earlier, okay?
No. Hello, George. So the weather increase in the temperature result in faster muscle growth? If yes, then global warming is a boon or bane?
Sorry, Prabhu, could you repeat that? The connection is poor. I could not.
Can I repeat again?
You kept breaking up. Yes, please.
Okay, sorry. Whether increase in temperature result in faster muscle growth? If yes, then global warming is a boon or bane? Still not clear? Do you think maybe I can change the question?
No, it's not. It's not the problem of the question. It's the connection.
Okay.
Can you hear me? Okay.
Yeah. I can hear you. Yeah.
Okay, so maybe I could just repeat back the question and see if that's what you asked. Are you asking if this improved muscle growth with temperature is in all species?
Kind of, maybe. I can maybe rephrase the sentence again. Okay. So do you think the global warming, like example, temperature can increase the muscle growth? Let's say it can increase the fish growth. If so, global warming is good for environment, like a boon or bane?
Okay. Well, no. Well, yes and no, because this improved muscle growth with temperature, it is not in all species. For example, in the example that I showed with the salmon, the fish that are incubated, or the eggs that are incubated at a lower temperature, at the cold temperature, they grew 20% better than the ones that were incubated at the 1 or 2, 3 degrees higher. Okay. in the case of Senegalese sole, then an increase of 3 degrees from 18 to 21 resulted in improved muscle growth. So it really depends on the species. Okay. Also, it depends on what kind of increase are we talking about? Is it half a degree, 1 degree, 2 degrees, 3 degrees? And then, of course, it's not necessarily a good thing if it will affect, for example, the immune system, or if it will affect the flesh quality, or something else that we didn't even look at. So we have to look at that with caution.
Okay. Yeah.
I would say that, you know, in this kind of knowledge can be used in hatcheries, like in more controlled environment, to manipulate the temperature without changing too many other things. And even that, you know, we need to look not just at muscle growth, but what other effects it has on, I don't know, like stress markers, other immune parameters, and so on.
Yeah. Okay.
Can be good, can be bad.
Yeah.
Like most.
Yeah.
Thank you very much, Dr. George, and thank you, Dr. Prabhu and Dr. Anush, for a wonderful interactive session. Just for the participants, we for other coordinators and organizers as well, we have kind of polling. We are going to display a few questions about the topic. You will be given, participants, you will be given 2 minutes time. Okay, we have kept some awards, best participation awards. So you please try to answer those questions. It's a multiple choice questions. You'll be given 2 minutes time. During that time, I request Dr. Esham Sivaprakash to get ready for his speech about the impact of COVID on fisheries. After that, he is going to introduce Dr. Ajad IS, and I request Dr. Ajad IS to get ready for his presentation. Thank you, both of you. Participants, you need to start answering.
Dr. Shantanu?
Yes, madam, please.
Dr. Ajad, he has joined?
Yeah, he is there. He's there. He's there.
Yeah.
Professor George, yours was a very interesting and enlightening talk. I really enjoyed it. Thank you so much.
Thank you. Thank you very much.
Can I say something? Jitendra, can you post the other questions? Only it's stuck in a question number 1.
Yeah.
Hello.
Yeah. Hello.
Dr. Shantanu, yes.
Yes, sir. Just for one minute. Okay.
Okay.
Yeah. So yeah, please get ready, sir. Thank you very much. Sorry for some question answer is going on.
No. I had just posted on your chat box and on Prabhu's chat box. Can I ask a question? I think you have not seen probably.
Oh, okay. I think chat box, we have some problem with the chat box. Could you please? Yeah. Sorry for that.
Dr. George, can I ask a question?
Yes, please. I mean, I don't know. For me, it's fine.
Yeah, it's okay. It's okay. Go ahead. Yeah.
Dr. George, you know, it's the genetic work or the genomics work, all these things, especially the gene expression work, they take actin as a housekeeping gene, right? In many of the studies. Yes. Is it expressed uniformly in all the tissues at all the stages, and in a uniform level? I have doubts.
No, I also had doubts.
That's a wonderful question, sir. We have beta actin and GAPDH, and all these genes are used as a housekeeping genes or reference genes. We always, claim that they don't express, but if you really look into that, maybe some slight variation between the tissues, something like that, or between the treatments. So we don't know. It needs a further, deep studies, I guess. Sorry for interrupting. I think, yeah.
Yeah, Dr. Judge.
And you should, I recommend that you use more than one reference gene.
Oh, yeah.
They're all changing a bit, but if you use more than one, then you are more likely that, if this one is going up a bit, maybe this one is going down a bit, and you end up getting a more stable reference if you use at least 2 or 3 reference genes. But beta actin, it used to be, and also G3PDH, GAPDH, those two used to be the most popular reference genes, but not anymore, I would say, because people have been noticing that they do change a lot with, I don't know,
I experienced that. That's the reason why I asked. And on a lighter note, why do we call muscle fiber?
Why do we call
muscle fiber?
I don't know, because it's actually quite, when you look at the muscle, if you make...
Yeah, it's like a fiber.
Yeah, like a fiber, almost like a rope, where you have this, almost like a cotton fiber or this, it's true. It has a very interesting structure, the muscle.
Yeah.
And can be very long as well. So maybe that's why they call them fiber. In the old days, they had a very, very good imagination, the scientists, to come up with names of things.
Yeah.
It's true.
I think the poll is closed now.
Sir, thank you. It is over now, sir. We can proceed, sir.
Okay, yeah. Thank you very much for all the participants who have participated in answering those multiple choice questions. And once again, I take opportunity to thank Dr. George, and I request Dr. George to stay with us for the next presentation with Dr. Ajad Ismail. And the next one, so Dr. S.M. Shivaprakash is going to speak about impact of COVID on fisheries just for a few minutes. Just I take this opportunity to introduce Dr. S.M. Shivaprakash. Shivaprakash, sir, are you there? Yeah, good. Briefly, I'll introduce. Thank you very much. And evergreen smiling person here, okay? I always say, and we try to do that one for happiness. Dr. Shivaprakash is a former Director of Extension Education, Karnataka Veterinary and Animal and Fisheries Sciences University, Bidar, Karnataka, that's in India. And also, he was a former Dean, College of Fisheries, one of the first colleges in India. It is in Mangalore, Karnataka, India. And he pursued his BFSc and MFSc, Bachelor of Fisheries Science and Master of Fisheries Science from College of Fisheries, Mangalore, and PhD from University of Mangalore, right? And after that, he pursued and he went for higher studies at University of Bergen, hope I'm pronouncing right. It's in Norway for a few months. And then he had a research attachment with the University of Stirling, or Stirling University. It's in the United Kingdom. And also he visited Dalhousie University, Halifax, Canada, for a training course on exclusive economic zone management. That's beautiful. I still remember you used to share your experiences with us during my studies in 95 to 2000. I cannot forget, and I will share those again. I'll remind, okay? And then he had several awards in his account, just to mention few: Young Scientist Award, and then Best Research Project Presentation, and then University Gold Medalist in Youth Festival. That's really beautiful. Okay, and apart from these, he also served as a member Board of Management at Tamil Nadu Fisheries University, Chennai, and then Secretary of Association of British Scholars That's in Mangalore. That's for Mangalore chapter. I wish to join that. I will discuss with you later. I'm really interested to join British Scholars chapter. And then he served as an external examiner at ASRB, and also he served as an external examiner for several PhD students. And apart from this, he also published several research papers and articles, mostly focused on aquaculture and fishermen community. That's really wonderful. And because of him, science reaches lab to land program as the best person for director of extension education I could see in COFSU. And then other than this science and academics, he's wonderful in composing poetry or poem. And not only that, he's beautiful in reciting those poems. And his portrait sketching is awesome. I have seen, and many of our colleagues have witnessed that. And wherever we go, that's a passion he has. And sense of humor, it is unbeatable. Okay. And then we wanted to invite you for our college just during the month of March, but we could not because of COVID. He wanted to share key to happiness and zeal for life. That's if you meet him, you will get how to lead a happy life. With that brief introduction, I take this opportunity to welcome Dr. S.M. Shivaprakash, sir. You are going to be live now, and please take over and speak about impact of COVID on fisheries. And after that, please kindly introduce Dr. Ajad for this presentation. Thank you very much, sir. Yeah, please.
Thank you, Dr. Shantan Gowda, and all the other faculty members of College of Fisheries, Ludhiana, Punjab. First of all, I would like to compliment Gadwas and College of Fisheries, Ludhiana, for having organized this very interesting international webinar. And furthermore, I would like to compliment the young faculty who have innovated aquaculture in saline soil conditions, where they demonstrated that Vanami can be grown in hinterland where there is saline soil also. So that is one thing it has opened up in Punjab because of the efforts of our young faculty of this College of Fisheries, Ludhiana. I compliment them because we all know that aquaculture is going to stay permanently to safeguard mankind. This is my belief. We all have studied fisheries science. We all are passionate about fish. So at the outset, first of all, thank you for having invited me, and you have told good words about me. Friends, at the outset, I would like to emphasize the importance of aquaculture in the form of a poem, because I believe poetry is a wonderful medium to communicate the message. What is told in 1 hour can be told in 1 minute. So here I go with my fisheries poem on aquaculture. Friends, maximize your imagination to realize what may be world's population after hundreds of generations. Think of the future life situation and shudder to imagine teeming billions, hunger and malnutrition. With shrinking fertile land and its destruction, making way for large-scale urbanization, can agriculture alone meet the future challenge of food production? Food technologists are of the opinion that fish is the ultimate solution to save mankind from starvation. If only we prevent environmental degradation, conserve existing fish population, and sustain global aquaculture production. Thank you. Friends.
Beautiful.
Yeah, thank you. Now, I would like to say a few words on the impact of COVID-19 pandemic that has on fisheries. We all know that the moment lockdowns started all over the world, fisheries also was a casualty, and there was complete ban on fishing, and there is complete ban on export. So all those fisher folk who were busy catching fish, all of a sudden they lost market. They lost distribution channel of sending fish to furthermost places. No air freights. So friends, this was first major impact on fisheries. And as a consequence, price fell down. They didn't know what to do. And then with that major problem started redundancy of labor. Labor who were engaged actively in fishing, they had no jobs. They were redundant. And then they migrated back to their native places. In lakhs, in hundreds of thousands of laborers, they went back to their village. And fishing industry went to hibernation. So these are the important impact we had on the negative side. Well, I would like to highlight the positive side. Friends, as all of you know, fish stocks are under stress. We have overexploited the resources all over the world. And as such, Canadian scientists have forecast that if we don't take ameliorative measures to prevent overexploitation, fish stocks in all the oceans would vanish by 2048. So, in the light of that, after the Code of Conduct for Responsible Fisheries was promulgated by FAO, most of the countries, more than 200 countries, agreed to implement those laws. But it had no teeth, but it had the spirit. And India also was a signatory to that. And as a result, our country, India, imposed a uniform fishing ban for 2 months. Earlier it was for 60 days. Earlier it was for 90 days. It was brought down to 45 days. And now it is restored to 61 days. 61 days along West Coast and 61 days along East Coast. Now I would drive home to the point of advantage or the beneficial impact of COVID lockdown. Friends, the first and foremost beneficiary is our fish stocks. For instance, in our Indian subcontinent, monsoon heralds fortune to the nation, fortune to our country, economically in all respects. This monsoon also augurs well for our fisheries because our fishes breed during monsoon time. During southwest monsoon, West Coast fishes breed in massively, and in the East Coast during northeast monsoon fishes breed. And in the light of that, now April, May, June, July, August, for the last 5 months, even this month also including, because sea is very rough, no fishing is possible. 5 months of complete closure of fisheries augurs well for the stock, for its revival. For instance, Indian mackerel, oil sardine, and most of the pelagic fishes, they breed during monsoon time, June to September. Now the point is, mackerel, for instance, Indian mackerel, it grows to commercial size, that is 18.5 centimeter in a matter of 4 to 5 months. They are known to grow very fast in their juvenile phase, and then they attain adulthood very fast, just 4 to 5 months. So fishes born in April, May, June, July, August, by August they are ready for harvest. Those born in May, they are ready for harvest in September. Those, like this, there is continuity of recruitment of fish into the exploitable phase of fishery. Now that during the last 5 months, there was no interference of man with the nature, that is ocean ecosystem, our fishes must be breeding happily. That is my proposition. And very quickly, I want to add, I have used an emotional card to say how important it is for us to conserve fish resources and observe fishing ban. My 4 lines go like this. Come monsoon, Indian subcontinent goes wet, requiring warmth and heat. but fishes have good time during monsoon with their honeymoon. Let the orgy of spawning go on, and none cast net on them. Let them procreate, recruit, and populate the already highly impoverished aquatic habitat and bring smiles on fishermen by landing in their net. Let the monsoon honeymoon go on and the smiles on fishermen stay on. So twin objectives of conservation and availability of fish for fishermen can be achieved by simply following this monsoon fishing ban. In fact, Baba DOM of Senegal has said, in the end, we will conserve only what we love, and we will love only what we understand, and we will understand what we are taught. So creating awareness is the first step, I would say. With that, we can be rest assured that our resources are in safe hands of our present day generation and future generations as well. So these are a few remarks I just wanted to make in the beginning. Shantan Gowda has given me 5 minutes. I think I reached.
Sorry for that.
Well, friends, it is my pleasure to introduce an illustrious scientist, Dr. Azad Ismail Saheb. It's my pleasure because I know Dr. Azad for the last 42 years. For the last 43 years, I know him. Dr. Azad also studied in the college where I graduated. He was 1 year junior to me. He completed his undergraduate in 1981 and he completed master's in 83, and then he completed his PhD in 2007 with the specialization of aquaculture with fish health and immunology. So he initially worked as principal scientist in ICR system, Indian Council of Agriculture Research in India, in different places, and he joined Kuwait Institute for Scientific Research in 2004 as research scientist. Since then, he has been serving in Kuwait. He has a total of 29 years of experience as a scientist. He has an illustrious career and has handled 15 projects as principal investigator, out of which 10 are at Kuwait. He is associated in 8 projects and has 4 patents to his credit. He has published 50 research papers in peer-reviewed journals. He has contributed 5 book chapters and has 26 popular scientific articles. He has published 15 extension publications, 5 manuals, and 27 project reports. Friends, during his 19 years of research experience in freshwater fisheries, marine and brackish water aquaculture, with emphasis on aquaculture fish breeding, a tribe which is a rarity. He is also an expert in fish nutrition and fish health management. Awards, I'll take only 30 seconds. He initially, for his master's and PhD, he received junior and senior research fellowship from Indian Council of Agriculture Research. And then he received Best Overseas Indian Fisheries Scientist Award for 2006 by the Indian Professional Fisheries Graduates Forum, Mumbai, India. Scientific Achievement Award for 2010-11 by Kuwait Institute for Scientific Research 3 times between 2011 and 2015. And Scientific Excellence Award for a US patent for 2014-15 by Kuwait Institute for Scientific Research. So these are the colorful feathers in his cap. I am very happy to introduce him to the world community. Thank you.
Yeah.
Over to you.
Yeah, thank you very much, sir, for beautiful, wonderful poems and for recitation and nicely introducing Dr. Azad IS. Okay, Dr. Azad IS, welcome. And you are going to be live now. And please unmute and share your screen and and take over the presentation. Thank you very much. Please go ahead, Dr. Azad.
Thank you, Dr. Shivprakash, my best friend. We used to be in together in dramas and so many other cultural activities. Though he was 1 year senior, it was almost like 1 year senior or 1 year junior, almost like classmates only in Fisheries College. So thank you, Shantan Goda. Thank you, Guru Angad Dev University for giving the opportunity. And thank you for especially the very good talk on epigenetics by George. And it's a nice opportunity for me to listen to him. Thank you, Shantan Goda, for that. And let me straight away go to the presentation. I hope you are seeing this.
Yes, sir. Yes, sir. Yeah.
I am getting a message. Your net connection is unstable or something. Please bear with me if there is some lag in the presentation. Shall I continue?
Yes, sir. Please go ahead.
Hope I am audible.
Yes, sir. You are audible.
You are audible.
Yeah. So the topic is vaccination. I have coined a word eco-host. Eco-host is ecological and host friendly. So eco-host friendly approach for sustainable aquaculture. Now, without giving much introduction, I just give a kind of overview. What could be the requirement of food? You can see here in the graph, the population is growing steadily, and the food supply curve is dropping now. It's going to be flattened now. So what will be the situation in near future? You should be able to imagine. Now, are you seeing any messages on the screen?
No.
No, it's fine.
No.
It should be...
No?
No.
Okay. Thank you. Okay. The present situation is quality but not the quantity of food, which is important. So FAO, according to FAO 2001, it says food security exists when all people, all times, have physical, social, and economic access to sufficient, safe, and nutritious food, which meets their dietary needs and also food preferences for active and healthy life. Taking this into consideration, we can see that fish is the best source of animal food and for quality food. Yeah. I think, yeah. What's the way out? We want to meet those demands. What's the way out? That's by enhancing food productivity, by enhancing food diversity, by exploring untapped resources, enhancing food quality, and reducing the quality reduction, that is post-harvest losses, and reducing the wastage of food, that is storage and also post-cooking, after cooking or distribution, whatever you call it. Now, to achieve this, the production of food, we had different phases in the Indian context, especially the Green Revolution, the White Revolution, and the Blue Revolution, which is, it's now starting to grow. That's why you can see a very thin band of the Blue Revolution thing there. Okay. Now, the Blue Revolution is aimed at producing food from the aquatic resources. Now, in this, we require a certain prerequisites for doing this meaningfully. Like we should have information on the speciation, the taxonomy, natural history, and biology of the species. So we are going to use them in aquaculture. We need to have information on the fish breeding, and we need to do improvement on that. And then we have to have methods of selective breeding, gene manipulation. Just now we had a talk on that. Then nutrition and live food biotechnology. This is for the fish to grow. And sex manipulation, as it has been already said that there is some problem in the maturation of the fishes or synchronization of spawning. And we are having similar problems with silver pomfret in Kuwait, where we have good quality females, but the males are not of very good quality. Sperm quality, as Dr. George has already told, sperm quality is not that good. And also the population of silver pomfret in Kuwait is not that much compared to India or the West Coast resources. Then we have algal production for the then enrichment so that we get better meat quality. Then we have ornamental fish or non-food aquaculture aspects. Then we have to have knowledge on toxicology because that interacts with the host and the environment more closely than any other thing. Coming to my particular area of talk, it encompasses microbial manipulation, bioremediation, especially in aquaculture context, disease diagnostics, and prophylaxis and vaccines. I call them as MIBD. You can see here. These are the 4 components which constitute MIBD component. Now, if you see different aspects of that particular MIBD, we have probiotics, then we have remediating agents like from the environment itself, we can have bioremediators. We have vaccines, bacterians, and recombinant antigens and things like that. Microbial vectors which can produce peptides or antigenic epitopes. Then we have a complex component called bioflocs, biofilms, and all those things. Nowadays, the biofloc technology is taking is getting into more importance in the aquaculture. Shantanu,
yes, sir.
There is one, this one coming. The people are waiting in the waiting room or something. I think it masks my this one here. Is it from my side or from your side?
No, for everybody, and we immediately admit them. Please coordinate.
No, that masks that covers my presentation. That's right.
Okay. Sorry for that interruption.
Yeah, it's okay. It's okay. It's okay anyway. Now the vaccine component, as you see, it is within a broader gamut of immune stimulators. The immune stimulators, the general idea was to generally stimulate the immune system so that it can effectively destroy the pathogen. Resistance in host, how long it persists, and prolonged administration, because we have to have a continued or sustained response. Okay. For bacterians and vaccines, the idea was specific targeting for specific pathogens. So it is the pathogen. The weaknesses, again, cost, regulatory constraints, limited knowledge, and not effective in the larval stages because larval stages are yet to be mature in terms of immunity or immune system. So that's why vaccines only. The reason is the multifaceted diseases or mostly the syndromes and things like that in aquaculture in the present day scenario. This can be managed if you have better management practices in place. Then intensification and state-of-art aquaculture will have an importance on the specific pathogens because they can assume we are using, say, like a RAS system or a complete biosecure system. If something happens somewhere, a loophole, and as a pathogen enters, it will completely destroy the system. So that's why we should have the vaccines in place. Then specific pathogen will have specific action. That's why vaccines we have to use. That's the answer. Potentialities of polyvalent vaccines have been proved. Of late, we have several polyvalent vaccines being demonstrated to be effective. So vaccines will play a very important role in aquaculture, especially in enhancing the productivity point of view. They have long-lasting effect compared to the other methods of disease management. Above all, they are eco and host friendly. Now, not going into the details of vaccines, their history and all those things, I'll straight away go into our experiences. My first experience was with the Aeromonas hydrophila, the biofilm of Aeromonas hydrophila. This formed my PhD thesis. The concept was like this. The problem was hemorrhagic septicemia, okay, in carps, which could be dropsy and also hemorrhagic septicemia. The hypothesis was oral vaccination with biofilm of Aeromonas hydrophila enhances serum antibody production in carp. Now that gave rise to several offshoot results like protection and reduction of losses, and biofilm is as a way of antigen protection, protected delivery for better mucosal and serum immune responses. I am talking this because it is from the point of view of oral vaccination, not like injection vaccination or the spray or bath vaccination, because those are all not applicable to all the stages of fish. So that's why we concentrated on oral vaccines. So biofilm was a mode of delivery which was better in eliciting mucosal and serum immune responses. And the deliverable was significantly protected fish. The salient features of this particular concepts were Aeromonas hydrophila is the major causative for the dropsy and hemorrhagic septicemia. Then bacterians and vaccines, those were produced that time, were not consistent in giving results, end results, what we wanted. And in this particular case, the carps, they don't have true stomach. So it's a good model for us to test whether the biofilm vaccines help in producing the required immunity. And intestine is very long in carps, and that is a disadvantage because we have to know where exactly is the antigen being destroyed, if it is the proteolytic degradation of the antigens and all those things. But however, we wanted to know whether the protected kind of antigen like the biofilm of Aeromonas will reach the immune responsive hindgut by bypassing the foregut or the stomach. Of course, in this case, there is no stomach. Then immune responsive region is hindgut. That's why we wanted to know this. Antigens tend to be lost as they reach the hindgut. That's one of the reasons why we wanted to know, take this as a candidate. Need to conserve antigenicity, that is of course is there. Capsulation is the answer, but the encapsulation of the antigen, it can be polymers and several things, but biofilm formation is a very natural thing. So we wanted to use this. But what actually happens in a pathogenic environment is also a clue for us to take up this particular experiment or the project. Because in pathogenic environment, the bacteria which are there in the pathogenic state in the system, in the organ systems, are not like what they are there in the broth or whatever in the lab condition. Like we grow them in the broth, we provide a lot of nutrients, so they grow normally and they don't have extra things added to that. Whereas in the biofilm form of growth, which they become pathogenic, they have certain extra things added to them. And that is called glycocalyx, and which is very resistant to enzymes and acids. And in our case, we have seen that it is resistant to even formalin and heat. We needed extra heat and more concentrations of formalin to inactivate a biofilm form of Aeromonas hydrophila. Biofilm antigens can be presented in right form and quantity because of this particular character, this particular property of the biofilms. And we projected a better immune response. So that's why we wanted to take up this aspect. This is the latest publication by Rebol. It's the concept, they have taken the Aeromonas hydrophila only, the concept of how it forms the biofilms. Probably the diagram is not very clear and the text is not very clear. You can just note down that's Frontiers in Microbiology 2017. This paper has come. So you can just go through because various mechanisms have been explained in this. Now what we did in this particular particular experiment was we isolated and confirmed the pathogenicity of isolate. Then we did culture them in a lab using TSB, the soya agar, used chitin flakes as the substrates because we have to provide substrate to the bacterium so that they can attach onto the surface substrate. And we use chitin because chitin can be used as a component in the feed. So we use chitin to form as a substrate of the biofilms. Then we assayed the growth dynamics of the biofilms for optimizing the densities. Fourth day harvest was found to be the best with 15% normal SB. Just to put you a point here, the biofilm formation is induced actually. Like if you provide all the nutrients, they may not go into a biofilm mode of growth. So you have to provide them some kind of hindrances. Like in nature, in a stream of water flowing over rocks and all, you can see some films formed. That's because of the dynamics of that particular stream. The organisms are not, they are washed away if they don't attach to certain things. So that's why they are made to attach onto a substrate. That is the flow is making them to attach, and flow and also the nutrients, because in a flowing system they don't get sufficient nutrients. So when they attach, they can trap more nutrients and then grow. That's the reason why the biofilm mode of action takes place in depleted conditions of nutrition. So that's why we used 15% normal TSB for growing the biofilms. And then we assessed the harvest for the population density. Then we inactivated them using heat and formalin. And we saw that formalin is better in eliciting immune response compared to the heat, because heat was disrupting many of the antigens which were antigenic. We resorted to formalin inactivation, then incorporated them into the feed at different microbial densities, then optimized the in-feed biofilm and versus the best antibody production and protective response. Then we assayed the antigen in different tissues using immunohistochemistry, and optimized the duration of vaccination for evaluating the protective response. The nutshell of what we found through these results were, of the 3 doses we used 10, 15, we used 10, 15, 20 days vaccination. 10 to the power 10 cells per gram of feed per fish per day was the best compared to 15 and 20 days. Okay, and sorry, 15 and 20 days induced higher compared to 10 days. So 10 days was deleted and we used 15 and 20, and statistical significance showed that 15 and 20 were not different from one another. So to minimize the use of vaccine, we resorted to 15-day vaccination. Among the 3 carps, we used Katla, Rohu and common carp. Among the 3, Katla produced the best results. And independent of dose and duration, antibody titers on protective response increased with time, and it went up to 120 days, and after 120 days the protective response started declining. Among the 3 doses, we used 10 to 7, 10 to 10 and 10 to 13. As I said already, 10 to 10 was the best, though 10 to 13 also provided the 10 to 10. You can see here one of the pictures on the immunohistochemistry results. The top 2 are the control results. You can see the vacuoles and the macrophage leukocytes, they are immunopositive. You can see a lot of within the tubule and the intratubular spaces in the kidney, and the villi you can see here, and the, within the kidney tubules also you can see, and the villi at the bottom right, and this is a similar slide with on the right side, on the left side, on the bottom, it's the villi. And these are our 2 publications, one in diseases in aquatic immunology. Coming to our second experience, which is, of course, this is the patent giving experiment or the project. Here we had the problem of Euronema, which is a marine ciliate. It's a holotrichous ciliate. It is similar to the itch or the cryptocaryon parasite. And this was the itch parasite or the marine itch Euronema parasite infections were mediated by Vibrio, which is naturally there in the marine environment. So we wanted to have a kind of vaccine which can counter both the Vibrio and the ciliate. Now, you know very well that the ciliate, freshwater ciliate Ichthyopteris multifilus or the marine ciliate cryptocaryon irritants, both of them require hosts for propagation. For making a vaccine, you require a medium where we can grow them in large scale. So providing a host is not a natural or very congenial thing for a large scale production of a vaccine. So we wanted to know what best can be done. Euronema, as you know, it's marine holotrichous ciliate similar to the cryptocaryon or the Ichthyopteris, and it's a bacteriophagic kind of ciliate, and it grows mostly on the bacteria. We have seen that Vibrio alginolyticus forms a very good food for this particular parasite. So we grow them in Vibrio soup, and after 4 days, when we could get a huge number of ciliates encapsulated with a lot of bacteria, we use them for inactivating and then incorporating in the feed. The hypothesis was oral vaccination with UVVac, we call them UVVac, enhances serum and mucous antibody production. And we used the bluefin bream, which is called Sparidentex hasta, which is a relative of European sea bream. We wanted to check the protection so that there is a reduction in the losses. And it's a way of controlled production of ciliate encapsulated within the Vibrio, containing the Vibrio. Then deliverable was significant protection to the vaccinated fish. Now the fish was Sparidentex, and the antigens were Euronema and Vibrio. Then antigen preparation was Vibrio encapsulated in Euronema. What we used was the fry from about 0.7 to 0.8 grams, 250 per tank in a one ton tank. And we did immunostimulation of the larvae, because I told you in the beginning that we cannot use larval stages. And in this particular case of bluefin bream, we have seen in our previous studies on the ontogeny of immune system, the immunity becomes mature after 14 days, roughly about 2 weeks or 2.5 weeks. So we did immunostimulation of the larvae from 0 to 20 days. And later on, we did the oral vaccination using 3 different doses that I will come to that later. Oral feeding was done 10 days, 20 days, and 30 days. And we did assaying of the antibodies and other things from 60, 90, 120, and 150 days after vaccination. And the protective response and the antibody responses were evaluated. You can see here the serum components and the mucous components. On the top, you see after the ladder towards the left-hand side, you have the serum components, and to the right-hand side is the mucous components. And it's quite quite a bit of mucous components are there compared to the serum components. And the bottom one is it's not very much there, but this is the Western blot analysis. You can see the light chain is being more immunogenic compared to the heavy chain. Heavy chain is at 67 kDa, you can see that. And you can see a very good kind of reaction in the mucosal immunity. These are the right side BG, 10 DG, 20 DG, and 30 DG. You can see there at 20-day group, 20-day vaccination group, you can see very good immunogenic response of the vaccinated fish. And this is the immobilization of the parasite. We collected the parasite and put in the micro wells, and then added the serum and the mucus to get how they get immobilized or not. Because it is very well known that these holotrichous ciliates get immobilized by the antibodies, that is through an eye antigen. That has been already proven in Ichthyopteris multifilius. So we wanted to know how much of immobilization takes, and relative percent immobilization on the right hand side. So it is it is almost same in the case of 20-day and 30-day. 20-day group and 30-day group. So vaccination is good enough to bring about desired changes. So no need to go for 30-day vaccination. And this is on the, that was on the previous one was on the serum, and this is on the mucus, using the mucus of immobilization results, and that was also similar to what we saw there. And you can see here we have done a 30 days, 60 day, and 120 days. You can see it is in the 20-day group, it is maximum say 20-day and 30-day group. Both of them are statistically not very significant, the difference between 20-day and 30-day groups. And this is regarding the gene expression. We did wanted to know whether the immunoglobulin gene is being expressed in a way what we see in the immunoglobulin responses, the ELISA titers. So we wanted to know how best was it. So the top one is the 10-day group, the bottom left is the 20-day group, and the right one is the 30-day group. That's why you can see there it is starting at 10-day, 20-day, and 30-day. You can see here the 10-day group is having two peaks, one at 20-day and another at 120 days after vaccination. Whereas the 20-day group is having a higher peak and at 60-day vaccination. So probably this gives an indication that when it stops dropping somewhere at 60-day post-vaccination, so probably we may have to go in for kind of a booster. Similarly, in 30-day group also, it is the peak is at 60 days, and the drop is very very short compared to the 20-day and 10-day groups. So probably the most optimum or the best way is to go for 20-day vaccination, where the drop is not very fast, and also the peak is similar to those in the 30-day group. And this was on the immunolocalization of the antigens, how far they reach. You can see here, the top is the control, and the A is the control. You can see a lot of intraepithelial leukocytes, a lot of them, and C, and you can see the immunoperoxidase positivity, D, and it is the second one is at 60 days, that is C and D 60 days, and E and F at 120 days. You can see a very clear localization and proliferation of the lymphoid tissue in the intraepithelial spaces. That is called lamina propria in case of intestine. And that was true even with the spleen and the kidney, and mostly associated with the melanoma-macrophage centers. And in some cases, like the intraepithelial or intratubular spaces, you can see the proliferation of the lymphocytes within the spaces. You can see the intertubular spaces. And it is also associated with the reticuloendothelial system, which is bordering the blood vessels. And it is well known that the macrophages are congregated on the reticuloendothelial system, and they form as the repositories for antigen supply for a longer duration. And this is a similar case with the spleen. Ellipsoids you can see here, and you can see the lymphocytes, and again the immunopositive reaction, and associated with the melanoma-macrophage centers. Even in the heart, we could see a lot of lymphocytes. This is the heart tissue. That is YAF. And D, you can see the lamina propria and the basement membrane. You can see here a lot of immunopositive reaction. And B is the kidney. You can see a few kidney tubules. It's a head kidney, and a lot of immunopositive reaction here. You can see a faint orange coloration here. Okay, and it is associated with the muscularis of the stomach also. That's one of the important things we can note from here. Muscularis forms a kind of repository for these antigens. Probably that is slowly yielding to systemic immunity, because mucosal and systemic immunity, there is a kind of class shift over later on. So that was one of the reasons why oral vaccines were discouraged. In the beginning, to tell that oral vaccine and they are short-lived, and this particular experiment shows that if you are prolonging the vaccination, and they can elicit systemic immunity, which is very good for long-term protection of the vaccinated fish. You can see here the enhanced survival. This is general survival of the beta glucan. Okay, it is the control group. You can see here the beta glucan group. Not much of a difference at later stages. You can in the beginning the dark B and all those things. That is A, B, C, D. If it is there, that is A, B is significantly different, and B, they are not significantly different. So beta glucan and 10-day group not significantly different. This is a general survival, not the vaccinated or the challenge survival. This is general survival in the tanks. It's not challenge. So even the general survival is also affected due to you can see the beta glucan enhancement from control group to the beta glucan group in the dark one. The conclusion from this particular experiment was beta glucan helps in enhancing the general survival of the subeti. Then 20-day vaccination is the optimum for antibody response and the protection. Mucosal and gut associated immunity was induced, which is what we wanted to know. And vaccination and beta glucan immunostimulation helps in upregulating the immune genes. The third one in this particular project is still going on. In fact, it is almost at the completion stage, and the part of this has already gone for the patenting. This one, it is in the preliminary stages of patent examination. So I will not be giving more details of this particular experiment, but the concepts and all those things which are there already in the general domain on the literature searches. So here the main problem was the viral nerve necrosis and cryptocarrion. I said in the second experiment we had Uronima as the example. Here we wanted to take cryptocarrion because cryptocarrion is causing a lot of problems in our facilities here. It is not only in our facilities, there is a commercial marine aquarium nearby. There also they had problems with the discus fish, the seahorses. They had silver pomfret on the display. All of them were affected with the cryptocarrion. So we wanted to take cryptocarrion as one of the candidates for developing a vaccine. So VNN and cryptocarrion were used as the vaccine candidates. And we wanted to use Bacillus species, which is already proved to be a probiotic in our particular case in Sebring. So we used Bacillus to carry the antigens of the antigen genes of these two pathogens. That is I antigen of the parasite and the coat protein gene of VNN. And protection was the aim. survival and protection, and probiotic bacteria to express these genes in situ, in the gut of the fish was aimed. The salient findings still now, Bacillus species can be used as a potential gene expression tool for carrying the antigens. The species what we have used is already known to be this one, and it expressed very well the I antigen and coat protein genes. And we used different densities, 10 to 5, 10 to 7, and 10 to 9 cells per gram of feed. And 10 to 7 was found to be the best. And we used 10, 20, and 30 days feeding. 20-day group is the best one, it's optimum for feeding. And we did the antibody assays also. You can see here in these graphs. It's at 90 days we see the best responses, the peak responses. Whereas in the second one also you can see the second group, which is the 20-day vaccination group, is the best, and it is significantly different from the others. So 20-day vaccination was concluded to be the optimum vaccination period with 10 to 7 cells per gram of feed. And this is immunoreactivity studies for tracing the antigens. You can see the lamina propria and the interlamellar spaces and the lamina propria. You can see a lot of immunopositive cells. We have done this one for I didn't get the results yet because of the lockdown. In fact, we completed this in somewhere in February, and the lockdown started in March itself in Kuwait, somewhere in the second week of March. So our results are yet to be finalized. In a nutshell, the summary of the mites, protected antigens, the first one. Protected antigens elicit better response. So biofilm of Pheromonas hydrophila can be a good vaccine candidate for carps. Then in the second case, it's UVVac or the Uronima vibrio vaccine, bivalent vaccine is very good, and we are trying to commercialize it also. This is good in protecting the sea bream from Uronima and the vibrio infections. And we have seen the cross-reactivity with also, and that was also very good. Like if in Uronima, it produced about 60 to 70 RPS of 60 to 70, relative percent survival. In cryptocarine, it produced somewhere around 50, 45 to 50. So roughly only 10 to 15% only difference. So still it can be cross-protecting against cryptocarium. In the third case of recombinant or gene-manipulated bacteria, it can be a very good candidate for vaccine delivery systems. In conclusion, vaccine research in aquaculture can yield potential measures of designing management strategies for maximizing aquaculture productivity. Thank you. Thank you.
Yeah. Thank you. Thank you
on
very
top
much.
it is written shukran. On top it is written shukran. Shukran means thank you.
Oh, thank you very much. Thank you. That's wonderful. Thank you very much for sharing your expertise on vaccines development in aquaculture. Actually, we are running short of time, and we are receiving a lot of messages. And Dr. George wants to have a question with you and for interaction session. Dr. George, please unmute and go ahead with your question. Then afterwards, we will take participants' questions. Only 2 questions. Okay, Dr. Ajad, question for you.
Yeah.
By Dr. George.
Yes, Dr. George.
Yeah, please.
Yeah.
Thank you very much for a very nice talk, Dr. Azad. I really liked the overview about the state of the art of vaccination and sustainable aquaculture production in the beginning. It was very nice. Thank you. My question has to do with the biofilm, Aeromonas biofilm that you are using or you have used as an oral vaccine. That again, I find very interesting. This idea of using a biofilm as an oral vaccine, because it will be much more stable than a regular bacterial suspension. But I was wondering, and this has to do with the limited immunological memory of fish compared to mammals. Like, how much protection and for how long does this vaccine give the fish? That is one question. And the other one that's also related is, do you know if IgT is involved in this protection, since you are looking at mucosal immunity?
Yes, thank you. In the case of biofilm vaccines, we have seen that after 120 days, there is a decline in the antibody production and even the protective response. There is a decline, but still the RPS levels were somewhere around 15 to 25. So anything RPS of 25 plus is said to be practically good. So probably at 120 days, we may have to give a booster. That's a kind of take-home message from that particular experiment. As far as IgT, I didn't study on those things in that particular thing. We did only the IgM and the mucosal immunity, only that. And probably I can have your point in view when I'm doing the assays in this particular case, where we are using the same this one for the oral vaccination. That's a good point, and should be, in fact, it will give a kind of dimension to the vaccination, oral vaccines, in exactly what happens in the system.
Yeah.
Thank you. Thank you very much.
Yeah.
Thank you.
Thank you. Thank you very much to both eminent scientists for your kind gesture of interaction. Okay, thank you. And we will take two more questions from participants. On behalf of participants, Dr. Naveen Kumar and Dr. Prabhjit will ask questions. Please go ahead, Naveen and Dr. Prabhjit, please.
Good afternoon, Azad sir.
Good afternoon.
It was a wonderful talk, sir. Particularly we have grown manifolds in aquaculture, but this disease is still a challenge, and vaccination is an alternative. So although Dr. George had asked similar kind of question, but one participant asked very interesting question. He asked that, like what we do in humans, we go for immunization at a very young age so that antibodies are developed and the disease does not affect at a later stage. Do we have such kind of, in future we can develop such kind of vaccines or mechanisms that we can immunize the young ones at a very young age in the beginning, so that we don't have a challenge, disease challenge of a major disease like aromas, hydrophilia and all. Sir, please throw light on it.
Dr.
Hello. Yeah. Hello.
Yeah. Go ahead, please.
I was audible, sir. Did you got the question?
Yes. Hello.
Yeah.
Sir, did you got the question, or should I repeat it?
Yeah, you said about the age at which.
No, sir.
Which vaccination has to be done, right?
No, sir. The question is that, like in humans, we go for immunization at a very early age, and there is a disease protection. So, can we go for immunization in fish also, so that we can protect it against a particular disease at a later stage?
Yes. Yeah, it's a question which which we wanted to use, especially when I was doing the biofilm vaccine, the experiment. The same time, there was another project going on the antogeny of immune system in fish, especially carps.
Okay.
Like when the fish becomes receptive to antigens, there are different things like immune system structurally when it is present, and when it becomes functionally active, and when it is immunogenic. There are three different things.
Yes, sir.
So we have to study that first. And to give, in my case, in that particular thing, we had done antogeny analysis of the sea bream, and we saw that 14 days onwards, they are able to handle the antigens and produce antibodies. So we cannot vaccinate before 14 days.
Okay.
That's the point where we can say that you can start vaccination after 14 days.
Okay. Fine.
Now, after 14 days also, there could be different types of antigens acting differently on the immune system. Like you know very well that the polio vaccine is given at a very young age and some other vaccine at later age, right?
Yes, yeah,
so
right.
Antigens also have a kind of this one. So probably this is the area where the fish immunologist should be able to proceed further. Like there are studies on the ontogeny of immune system, both structural and functional ontogeny. So probably that is the cue for using when to start the vaccination.
Okay.
And again, booster is another question. What answers your question? So when to be given a booster? As I said in my earlier case, at 120 days the antibody response drops down. Okay, so probably that is the time at which you have to give booster. And maybe in some other cases, like in my second experiment, we have seen at 60 days it drops.
Yeah.
So we will have to give booster at 60 days. So in that particular antigen, we have to give at 60 days, whereas in the previous one, it was at 120 days. Probably the difference is the first one was a biofilm vaccine, which tended to be in the system for a longer time, compared to this one, where it was eliminated. Maybe the antigen is eliminated. That's why the antibody titers came down. So we have to give booster for one at 60 days and for the other at 120 days. So these are all the strategies which we have to work out by taking into account the ontogeny of the fish, particular what we are working, and it differs. Like for the carps, it is different, and for the marine fish, it is different. For the temperate fish, is different, or for the cold water fish is different. In fact, in cold water fish, and some of the fishes, it is 50 or 60 days. The immune system is not mature.
Yeah, thank you very much for a nice response. Naveen Kumar, you please go ahead with your question on behalf of participant, please.
Yes.
Thank you.
Azhar sir, it was a wonderful presentation. We got a very good information out of that 3 experiments, and very specific, because we got too many questions, and very specific to oral vaccination, because the oral vaccination is best for the aquaculture. And based upon your 3 experiments, have you find any tolerance effect in the fish immunity against the vaccines? Because we have seen in veterinary and humans. Have you seen in our fishes?
No. Because the tolerance effect we have to see by going for a longer period of the experiment, like 3 years or 4 years project. If you take up and see at go on giving boosters, and first of all, the major basic thing is you have to know the dynamics of the immune response without giving a booster, after giving a booster, and at different time intervals. Then only we can come to know when it exactly becomes resistant. So probably that is the place where we are lacking information. So in my experiments and all those, I didn't get any resistance, especially the second experiment. We are still having the fish with the vaccinated fish. Last year we had cryptocarrier infection, and we exposed them to the cryptocarrion. We got still very good protective response in those fish, but of course not comparable to the previous exposures. So still we had very good protection, but I cannot say that they became resistant. So we have to be very careful in telling whether it is resistant or not.
Thank you, sir. So just continuation to this question.
We are running short of time.
No, just one question. Have you found any side effect of this vaccination? Side effects, like we have seen in polio vaccine, 1 in million we get a paralytic polio. So same way, have you seen any side effects in among these 3 vaccines? What you have used in the experiment?
No. In my case, because we didn't use any adjuvant, number 1. Because adjuvants are those which are causing problems. And adjuvants in, especially in the micrococcus with lipid and all those things, they produce a lot of nodules, ulceration and things like that. So we didn't use any adjuvant. That's one of the things. And in biofilm vaccines also, we didn't find any side effects. Whereas in the last one, it is not the side effect, it is the pro effect. Like the last experiment where we used the probiotic. Okay, in that, after certain up to certain time, the probiotic effect is there, and after certain time, the probiotic effect is gone. So probably to keep both the probiotic effect and the antigen delivery effect, we may have to use the batch of bacteria which produce and express antigens, at the same time keep the probiotic property intact. So probably you have to use those bacteria at that particular time. If you continue that bacteria of the same batch for a longer period, probably you will lose one. So I think that's the knocking off mechanism of the genes probably after a certain time.
Yeah, thank you very much, Dr. Ajaz sir. And now we are actually running short of time. I'm getting a lot of messages. Dr. Prajvit, you want to go for a quick question?
A short question, which is very much related to this COVID pandemic. Sir, one participant was asking that, like it is presumed that this coronavirus, it came from bats. Out of your experience, can you tell us, is there any threat of any pandemic from fishes? Is there any dreaded zoonotic disease which can spread from fishes, aquatic organisms to humans and can take a shape of a pandemic?
I don't think so. Being a fish lover, I don't want to.
Okay, fine, sir. Thank you.
I don't think. I have not seen. In fact, there were rumors that earlier when we had the VNN problem, and there were a lot of fish kills in Kuwait. They wanted to ask whether this is due to the virus. In fact, it was all because of the anthropogenic activity. A lot of activities in the beach areas and all those things. And also, the temperatures go up to 55. Now it is more than 46 or something.
Oh my.
So water temperature 37 to 38. Very high. In fact, some 3-4 days back, the Hamur broodstock or the grouper broodstock, they stopped eating because the temperature was 36, 37. So immediately we had to start the well water, which is low in temperature. So those are all the things, and definitely no pandemic from fish.
Thank you, sir.
Thank you very much for a wonderful interactive session, and it's a very nicely taken up the questions, and your response was highly appreciated, Dr. Ajad. Thank you very much. And now we'll move on to concluding remarks. Actually, we expected our Director of Research, our university, Gadwasu, and Dean, College of Fisheries. Unfortunately, they are really busy with opening up of a laboratory for testing COVID here in our university. That's a great thing for our university. Now we'll move on to concluding remarks. We have Dr. Meera, head of the department, College of Fisheries, Aquaculture department, and Dr. Sethi. First, I request Dr. Meera to briefly give concluding remarks, and afterwards Dr. Sethi will take over. Dr. Meera, please go ahead with that. Thank you very much.
Thank you, Dr. Shantanu. First of all, I must say that both the sessions, they were highly informative, interesting, and highly interactive. And I congratulate all the speakers and the organizers for this grand success, that all the participants, they were actively participating, interacting with the speakers, and both the presentations were highly informative and well delivered. The message has been well received. And we hope that in future we will be certainly planning for fruitful collaborations. And if I talk about epigenetics, after listening to Dr. George's presentation, now I am wondering that how I can take up this field to see that how epigenetics can help us in improving production, or can it play any role in broodstock management, flesh quality enhancement, or disease resistance, or even manipulating sex ratio also. So certainly Dr. George, in future, we will be approaching you, communicating with you to develop some collaborative research and submitting some collaborative proposals also. I hope you will be providing us all the assistance and guidance to move forward. So thank you so much, sir. It was very good to have you with us in today's webinar. So secondly, Dr. Ajad, he shared with us his experiences regarding vaccine development for aquaculture. Earlier, I could not interact with him. I think he was a bit late in joining this session.
Yeah.
So, yeah. So it is our, you can say, honor to have you, sir, with us in today's webinar. It was very nice hearing to you. It was your talk was very informative, and it has enlightened our minds and our vision to take up this kind of research in future. And I would like to tell you, some of our scientists, they are already working in this direction. As you know, Dr. Naveen Kumar BT is there. He's working on these biofilms. And in future also, we will be keep on asking or keep on looking forward to you, sir, for some inputs from your expertise so that we can do better in this field. So it was very nice, sir. And I would like to thank Dr. Shiva Prakash also. His presentation, although very brief, but his motivational poems and message to see positive side of COVID pandemic was, it is really commendable, sir. So it was very refreshing to listen to you, sir. And during this session, everybody must have noticed that we had Dr. Eknath with us. So it was a big, you can say, it was our pleasure to have him with us. So thank you, sir, for joining this session and interacting with us, with the audience also. So I will not take much of your time now. We have Dr. R.S. Sethi with us. He's head department of biotechnology from College of Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University. He is one of the collaborating partner in organizing this webinar. So once I thank you all the speakers. It was a wonderful time listening to you all. Now I request Dr. R.S. Sethi for his vote of thanks. Thank you.
Please briefly give concluding remarks, Dr. Sethi, and then give word of thanks, please. Thank you, Dr. Sethi, sir. Dr. Sethi, are you?
Thank you, Dr. Shantanu. Thank you very much, Dr. Meera. And before I, formally extend vote of thanks, Dr. Meera, I fully agree with you. You have concluded, this webinar in a wonderful way. I have, few things to say. This COVID-19, it has taught us, not us only, even to the layman, there are two things they are talking about. One is the real-time PCR. They know what is it. And even they know importance of vaccine. Everybody is looking where the vaccine is coming. And people know which type of machine can tell if I am COVID positive or negative. So I think from the molecular biology or the biotechnology, it's a good thing from this COVID side. And when we talk about epigenetic studies in these time of days when the scientists are talking about One Health initiatives, these sort of studies become very important, and we are very much thankful to Dr. George for enlightening the audience on this very important aspect. Same time, we are also thankful to Dr. Ajad. He has updated the audience with his experiences. This become very interesting when someone talks about his experience and narrate the story. So the audience become more interested and we learn so many things. We liked your way, sir. Thank you very much for sparing times and to be with us. And above all, we are very much thankful to our Vice Chancellor, sir, for allowing us to organize this webinar. Similarly, Dr. JPS Gill, Director of Research, he could not attend today, but he was always helpful, starting from the beginning and providing every sort of support. Similarly, Dr. Kalbir Sandhu, the Dean, College of Fisheries, He was very, instrumental, and he taken care everything so that this become a successful webinar. We are thankful to you, sir. Then, I will be falling short of words if I do not extend thanks to our team. Everybody, the coordinator, Dr. Vineet, Dr. Prabjit Singh, Dr. Anuj, Dr. Naveen, Dr. Jitendra, Dr. Shantana Gowda, and especially, Dr. Prabhu Gowda. He did both were, everyone was, played a perfect role wherever it was required and to execute this planning in a wonderful way. Similarly, if I, say something about Dr. Meera, she will not probably like it, but it is my duty to say that she, supervised every small thing. Yesterday, she had a, online meeting of how the things should move, and she put lots of efforts. Thank you very much. And last but not the least, all the participants, we got a huge number of participants. Maybe we are missing someone. We could not allow to, admit certain participants. You could see, always on the screen, there were some requests coming up. But, this was our first webinar, and we were not, we can say that we did it excellently, but we were not prepared that much probably. So we ensure that the next time when we will be doing, we will be, allowing more participants. There are certain online limitations, restrictions. So thank you very much for to the participants to make this webinar a very successful one. Thank you, everyone. Most of the time at the end of vote of thanks, we say, let's have a cup of coffee. But, today I cannot say that. But thank you very much.
Yeah, thank you very much. So you thanked everybody. So it's my duty to thank you, Dr. Sethi, for organizing, being part of this organizing committee. You are a wonderful gentleman. Thank you very much. And now we have a polling session. That is, we have questions for the second session. So participants, please answer those questions. It's a multiple choice questions, like how you did for the first session. So now, Dr. Jitendra, could you please display those questions? So we have, it will take a couple of minutes. Then afterwards, you are done with that. Okay. Thank you very much, Dr. Jitendra. Over to you now. Yeah. Yeah, your question, first question starts now. You have, I guess, 30 seconds.
Dr. Azad, just I wanted to know one thing, sir. It's not a question. On your very first slide, the word "thank you" was it written in Urdu or Arabic? Because was it Urdu or Arabic, sir? Just I wonder.
Last one, right?
You write "shukriya." Yes, that was Urdu or Arabic, sir?
Shukran. Yeah, it is Arabic.
Okay, fine. It's somewhat like Urdu because, sir, I can read Urdu.
It is, yeah. It is same.
Okay.
Very few letters are different. It is the letters are same. Yeah. Okay. It is same.
Okay. Thank you, sir.
Welcome.
Yeah.
Shantana Gowda.
Yes. Somebody.
Shantana Gowda.
Yes, sir. I'm there.
Can I talk to Prabhu Gowda?
Yes. Go ahead with it.
Norwegian.
Yeah.
Oh, hitar do. Urdu kamar fra. Urmange klokka. Thank you.
Maybe you can speak to Dr. George.
Oh, yeah. Thank you.
Yeah.
My stay in Norway for 1 year for my PG diploma was a wonderful memory. Norwegians are very, very warm people. The cold country, but warm people. Wonderful. I learned skiing there. Even today, I am ready to go on skiing. See, some of the excitements that you had, you will never forget. Thank you.
Yeah, that's for sure. Thank you, sir. Just for participants, unfortunately, we could not open the chat box due to some technical error. Sincere apologies for that. That's why we just opened all the WhatsApp groups where you can give your questions. So your questions might not have been taken up, but your questions will be considered for best award participation we'll consider those questions for evaluation purpose. Okay.
And Shivaprakash sir, I just forget. Thank you very much. During this Corona stress, your songs on Facebook, they have been just relieving us from stress, sir. Wonderful voice you have.
Yeah.
Particularly those Kishore songs, sir.
Yeah, thank you.
Yeah.
I get fascinated by the lyrics.
Yes, sir. You sing wonderfully, sir.
Thank you very much. Thank you.
For all the participants, you need to give a feedback, and there is a link available there. It is displayed on the screen.
Dr. Shantanu Gowda, it went on very nicely, without any interference, without any technical errors. Very nice.
Yeah, thank you very much.
No echo, nothing.
Okay, thank you.
No, I should thank Shantanu Gowda for introducing me to Dr. George, and I am seeing Dr. Eknath after a long time. In fact, in SIFA, I think I saw him, or maybe in Cochin, I saw him when he came for this one, the fisheries forum. After that, no.
Very happy to
though I traveled many times to Bangalore, but I did not see him.
Yeah. Okay.
And introduction to Dr. Meera, Dr. Sethi, it's very good. Dr. Prabhjit. Prabhjit, how do you pronounce Prabhjit?
It's fine, sir.
Prabhjit. Yeah. So it's a nice thing, and I'm seeing a lot of them. Naveen, I didn't see him last time, I think, when I went. Did you attend last DAA conference?
Yes, sir. Yes, sir.
Okay.
I think we are done with polling that questions and answers and all. I'm very gratitude that you expressed thanks, and I'm sorry for keep on sending messages to you, emails and WhatsApp for getting information. Thank you very much.
That's required.
Yeah,
that's required, because this you are not seeing and then talking. No, it's all ye.
Yeah.
So ye means there may be some cut here and there, so you will lose that information. You have to keep on doing that.
Yeah. Thank you very much. I think hope we are done. Yes, sir. Yes, sir.
Starting one word, I just want to add.
Yes, sir. Yes, sir.
Our fisheries is under pressure because of the modern technology.
Yes, sir.
So there has to be a balance. If technology dominates, ecology suffers.
Yes, sir.
So here are two lines.
Yes, sir.
Technology versus ecology.
Yes, sir.
Marine catfishes got annihilated by porcine technology. Juvenile fishes got perished by trawling technology.
Okay, sir.
Brood fishes got killed by dynamite technology. Okay. Squids got diminished by jigging technology. Indeed, all technologies owe ecology an apology.
That's fantastic. Okay, thank you very much, sir. Okay, now with that we are done, and I request participants to go without what you call feedback. Thank you very much. Hope you enjoyed, and hope you got a take-home message from two great or eminent scientists from different parts of the world. Okay, and they enlightened with their expertise in epigenetics and vaccine development in aquaculture. That was a great session we had, and apologies for taking more time, and it was a wonderful and fruitful session. And I thank all the organizers, coordinators, and participants on behalf of College of Fisheries and College of Biotechnology, Guru Angad Dev Veterinary and Animal Sciences University. Thank you. With that, with your consent, we are going off now. Okay, is it okay?
Thank you.
Yeah, thank you very much. Thank you. Done. Yeah, thank you. Yeah. Okay, thank you very much, participants.
Bye, sir.
Yeah. Dr. Jitendra?
Yes, sir.
Yeah. You can do that one. Okay. Control it.
Okay, Dr. Shantanu.
Yes, madam. Yes.
Okay, congratulations for
Oh, yeah,
completing the task.
Yeah.
And congratulations to the whole team. Well
Yes,
done.
yes. Yeah,
Keep
thank
it
you.
up.
Yeah,
And
thank you, madam.
hopefully we will be organizing another webinar very soon.
Yeah,
Okay?
yeah, sure, sure. Yeah, thank you.
Okay.
Yeah,
Okay.
yeah.
Chalo, all the best.
Yeah, thank you.
Okay.
Yes. Jitendra? Yeah, please.
Congratulations to all the sirs and madam.
You can.
Okay.
Can you control that, Jitendra? Yeah, please.
Okay, Goda.
Yeah. Oh, thank you.
Congratulations. So
Yeah.
we can take a leave now, or there
Oh,
is
yeah,
some
yeah, yeah.
coffee,
I'm just
coffee
waiting.
or something like that.
Yeah, yeah, yeah. I
Jitendra,
understand.
please stop that YouTube.
Yeah.